Good morning, everybody. Welcome to the third and final day of the RISE Symposium. I'm going to be your host, Nitra, the STEM Connections Coordinator and the RISE Coordinator for the summer. Sorry for the little technical delay, but we are up and running, and we will start off again with our intro videos. This is, again, something I've been looking forward to every single morning. Thank you very much to all the faculty, staff, and students who've made this possible and put in a lot of great work. And we'd love to introduce today's speakers and presenters. All students were requested to submit a short 5 to 10 second video, just introducing themselves, their major, and also where they're from. So we get a little visual taste of where everybody seems to be working at nowadays. Without further ado, I will let Marissa take it over, and we will start our presentation or our program. Thank you.
Hi, my name is Manuel Bárcenas. I'm a mathematics major. I'm an incoming transfer student, and I live in Boyle Heights.
Hi, I'm Joshua Wong, and I'm a second-year biochem major, and I'm back home at the Bay right now.
Hey everyone, my name is Enrique CNAS. I'm a third-year mechanical engineering student here at UC Riverside, and I'm originally from Highland Park in Northeast LA.
Hello, I'm Imelda. I am presently doing my master's in public health at Charles University. I live in Riverside, California. Thank you.
Hi everybody, my name is Abigail Osunde. I currently go to Riverside City College. My major is bio and I live in Riverside.
This is Noor Hamsi. I'm a biology major and I live in Redlands. I transferred from RCC to UCR this coming fall. Bye.
Hi, my name is Taylor Oliver. I'm currently a general bio major at RCC and I live in Riverside, California.
Hi, my name is Natalie Masai. I'm a biology major going into my third year at UCR and I'm from Covina.
Hi, my name is Jonah Brunson. I'm a chemistry major going into my third year at UCR and I live in Riverside.
Hi, I'm Zara Saeed. I'm a biology major. I'm going to my junior year and I live in Norco.
My name is Kirsten Bailey. I'm a first year transfer microbiology student and I'm from Highland, California. Give me a shake. Good boy.
My name is Luis Velez. My major is cellular molecular developmental biology. I will be attending my first year at UCR as a junior and I live in Corona, California.
Hi everyone, my name is Megan Woods. I'm a chemistry major. I'm from Fullerton College and I'm currently living in Fullerton.
Hi, I'm Brian, rising junior, cellular molecular developmental biology major, and I'm in Riverside.
Hi, I'm Carolina Acevedo. I'm a biology major at UCR. I'm a senior status transfer, and I'm from Moreno Valley, California.
Hi, everybody. My name is Kiani Kosek. I'm a biochemistry major and an incoming junior, and I'm currently living in Hallandale, California.
Hello, everyone. My name is August Perales. I am a biology major, and I am from the Coachella Valley.
Hi, my name is Eric Torres. I am a biochemistry major. I will be transferring into UCR and I hope to graduate in the year 2022, and I live in Riverside.
My name is Dahlia Roscoe. I'm a microbiome major going into her third year of schooling and I currently live in San Bernardino.
Hi, I'm Jessica Black and I'm a third year neuroscience major.
Hey guys, my name is Lawrence Taraden. I am a third year biology student and I live in Moreno Valley.
Good morning everybody, my name is Chris Gadula. I'm an incoming second year majoring in biology and I'm from San Diego, California.
All right, thank you very much. That was a great introduction. We had a few people who also had presented earlier in the week, so that's why you may have seen a few students who we've even seen previously. In any case, we started a little bit late, so I'm going to jump right to it. This morning, we're going to start off with our student researcher testimonials. These are students who were both nominated and volunteered to speak more about their personal stories and also how RISE has impacted them over this particular summer. It's a very challenging time, and we've had many different students rise to the occasion and do very well. So without further ado, we have our very first speaker, Mr. Enrique Aguilar. Enrique, thank you for turning on your camera. And a very great scene. I see you are at UCR reporting at the engineering college. I'm over at the hub, so come say bye right after, or come say hi right after.
We'll get some lunch afterward.
Sure. I'll see you in a bit. The floor is yours. Thank you.
All right. Well, good morning, everyone, and thank you for joining us for this last day of the RISE Research Symposium. My name is Enrique CNAS, and I'm a third-year mechanical engineering student at UC Riverside. I transferred from Glendale Community College earlier this year in January. And this summer, I had the opportunity to work in the OSCON Energy Lab. I performed work supporting the development of next-generation lithium-ion batteries. Initially, I spent hours reading research papers trying to understand concepts that were new to me. I managed to develop programming skills necessary to analyze large amounts of data, and I will speak a bit more about this later on during my presentation. But for now, I want to give a little backstory regarding my involvement with Dr. Ozkan's lab. So like I mentioned, I arrived at UC Riverside earlier this year in January, and 3 weeks after arriving at UCR at a completely new campus with practically zero connections, I asked one of my professors, Dr. Cengiz Ozkan, if there was any work available in his labs. He directed me to speak to one of his PhD students, Tanner Zarin. And by week 10, I had been welcomed into Dr. Ozkan's energy research lab. Tanner went on to become a mentor of mine, and I'm grateful for the support of Tanner Zarin. He spent a lot of time teaching me everything that I needed to know to be able to perform my duties. And we would meet multiple times a week, and we would stay in contact for issues that arose on a weekly basis. And furthermore, becoming a RISE member has undoubtedly been one of the best decisions that I've made during my academic career. When I first signed up for RISE, I did not expect to feel such a sense of community, despite this being a completely virtual event. I expected to simply be sitting through weekly meetings and not doing much as far as participation goes. But it turned out that the RISE Summer Research Program was the complete opposite. Every week was filled with team mentor meetings, guest speakers, and social nights, and STEM office hours. And as a student who is still living near the university, I was extremely excited to be on campus all summer in my lab, but unfortunately, that was not the case. It was not an option due to the pandemic. However, RISE managed to take an otherwise unbearable quarantine summer and made it significantly more bearable. The weekly RISE events were something that I look forward to every week, and I'm extremely grateful for all the work that the RISE coordinators and mentors have done. It is amazing that they have been able to take this typical RISE experience and make it completely 100% virtual. It could not have been a better experience, in my opinion. In particular, I would like to thank Nhi Tran, Dr. Brandon Brown, and Marissa Carranza for coordinating and meeting with us on a weekly basis. Their wisdom and advice will prove to be invaluable. Thank you for your time.
Thank you very much, Enrique. We have our next speaker, Ms. Keone Kosek. Thank you very much for turning on your camera.
Hello, thank you, Nhi. Hi, everyone, and good morning. I'm Keone Kosek, a transferred student from Victor Valley College and a junior majoring in biochemistry. And this summer, I got the opportunity to do research under Dr. Don Nagel, an assistant professor of genetics in the Department of Botany and Plant Science. Being a part of the Summer Bridge program has been nothing short of a great learning experience for me. I've been given a lot of information regarding the resources and programs offered on the campus and now online. I found it to be beneficial to me because now I know what steps to take if I want to extend my academic and social ambitions. But when I first found out that I could still do research this summer, I had a mixture of emotions ranging from excitement to participate in an internship, curiosity since I'm a transfer student and this would be my first interaction with UCR, and I didn't really know what to expect. And there was also the satisfaction that I'd be staying productive this summer instead of just waiting around for my first quarter to start. But this program has given me more insight into the different areas of research offered at the school, and has made me anticipate immersing myself more into research itself, since it's a personal goal of mine. I didn't have a specific area of research that I for sure knew that I wanted to pursue. Instead, I like to think that I'm fairly open-minded to all areas. Although I never really considered research in plants before, I was really looking forward to studying them as well as understanding the larger role they play within our lives. Another benefit that I gained from this program is the practice it's given me to share scientific literacy. And that's a topic we discussed at one of our Zoom meetings. I feel like the research itself is important, but being able to take all that you did and studied and present it to an audience in a way that they will understand holds more importance. It's a skill that I'm still working towards, but I plan to use it throughout my academic career here at UCR and further. As for my lab experiences, I wasn't able to run any of the experiments in the lab myself, but my faculty mentor, Dr. Nagel, rose to the occasion and found a way to bring the lab to us. She made videos and walked us through the photos she took of the experiments, so that way we still have that visual representation of what was being done and how the machines worked. We also had side assignments that broke the ice between me and the rest of my teammates while we worked together. But of course, it also kept us busy while Dr. Nagel ran the experiments. Overall, it made for a very interactive experience, which I thought would have been difficult to do with us being behind a screen. Even though it was all remote, some of the things I learned how to do was read DNA sequences, perform statistical analysis, and how to annotate genes. But of course, it did come with some challenges. For me, mainly, it was being able to remember all the steps for the protocols of our experiments since I wasn't physically there to do them. And our protocol was fairly lengthy with repeats. I often forgot and was confused as to where some of our products from the reactions came from. But to overcome that, I did write out the steps in my lab book to keep track and to understand why each step was necessary before we could move on to the next. And I also had my teammates and my faculty member there to bounce my ideas back and forth, and that was very reassuring to me. But as I stated earlier, our faculty mentor also compiled all the photos and videos of each experiment together, so we could always go back and reference them whenever we needed. As for my teammates, this really isn't a goodbye for us. It's more of a see you in October since we all share the same class together. For future participants, I would tell them to not be afraid to apply and to go out for research opportunities like this, because I like to live by this notion. It's better to apply and to know than to not apply and not know how far you could have gone, because it really pushes me to go out of my comfort zone and explore areas that I would have otherwise passed up. In the beginning, it may seem daunting and confusing, but it's supposed to be a learning experience, so don't feel like you have to know everything right away. And with these last words, I will say thank you again, RISE coordinators, peer mentors, and faculty mentors for your endless words of encouragement and support during these past 8 weeks. I've enjoyed all the research I've been able to conduct, as well as the people that I've met in my lab meetings and during the social nights. Surprisingly, I've never played nor knew what Kahoot was up until now, and now I understand the hype around it. This has not only been a memorable experience, but it's also helped me feel like an official UCR student. And I also want to give a special thank you to Marissa for sending us the care packages as well, because they were very much appreciated. Thank you.
Thank you very much, Keone. And last but not least, we have our third speaker and final speaker for the morning, Ms. Kirsten Bailey. Thank you very much. Oh, hey, Kirsten, I think we'll need you to unmute. No worries.
Sorry about that.
You're okay. Take it away.
Right. Once again, good morning, everyone. I'm Kirsten Bailey. I'm a transfer student from San Bernardino Valley College, and I'm going to be an incoming junior and microbiology major this fall. I was accepted to RISE through the Summer Bridge to Research program, and I was a part of Dr. Mock's lab in the entomology department. Our research was funded by Dr. Purcell's Six Lakes grant, and I'd like to share more about my online experience with RISE this summer. When I first got my acceptance email to the program, I think like everyone, I was really excited because I've never been a part of research before. However, I was quickly realizing that I was going to miss out on all the fun physical aspects of the research internship on campus. However, this summer has surpassed any expectations I've had. RISE has really helped me align my time outside of the virtual classroom with my future goals of eventually working in a clinical lab. I've always pictured myself behind the scenes, like doing something like this experience would give me, but I've already learned so much how to implement all these tools I've gotten for my coursework preparing for this fall. Orientation was 2 whole months ago, and it showed me that I was going to be a part of something truly helpful to my college experience. Our brown bag meetings with Dr. Nugent were invaluable, and I learned so much from those sessions, how to be a better science communicator and how important it is to be able to communicate what we need. I've learned so much about my possible paths after graduation that it wouldn't have been possible without me, Marissa, all our peer mentors, as well as fellow RISE participants. I learned how important it is to use Google Calendar and all the programs we can join this fall. My RISE participation made registering for classes seamless, and I even started a group chat with other microbiology majors to share how much I was learning this summer and all the easy ways to add classes. Our peer mentors felt like our personal guides to uncharted territory. They did an amazing job keeping us involved, and we all had a great time doing it, I think. They also answered all of our questions, all times of the day, and I really appreciated that. I also want to thank Dr. Mock. I learned so many new ways to communicate, and learning how to communicate with an instructor or a professor, I think is going to really come in handy for the rest of the year. So it's been really great interacting with everyone this summer. And I want to thank my mom, my sister, my family for asking questions and being interested in what I'm doing and really being there for me. So I appreciate being a part of this. Thank you, guys.
All right, thank you very much, Kirsten. Great way to start off the morning from all three of you. We have now we're going to start our oral presentations. And our oral presentations, as a reminder, are approximately 12 minutes. They are complete PowerPoint presentations. And there will be about 2 to 3 minutes worth of questions after each presentation. If you'd like to ask a question, please feel free. We will take questions through the webinar chat that's open here, or you can raise your virtual hand on the panelist or the participant option and you can ask live. We will simply unmute you. And then we will move on to our additional ones. But our first presenter, Mr. Manuel Barcenas, is available. Thank you very much. I see you are also at UCR. I think it's a very popular option this morning.
I had to make a quick trip.
Thank you very much. We appreciate the effort. So your PowerPoint is on the screen here, and you may go ahead and take it away. Thank you.
Thank you, Nee. Hello, everyone. My name is Manuel Barcenas, and today I will be discussing the computational modeling of airway epithelium lineage. So we all know that breathing is important for most life on Earth. When breathing, the airway epithelium, denoted by the black box on the figure, acts as a physical barrier between the body and the environment. But what happens if the airway epithelium suffers damage? How would the airway epithelium, or AE for short, heal from this damage? Well, we can already see that a model would be useful in understanding just how these AE cells react to damage. So here is the outline for today's talk. First, I will talk about the biology of airway epithelium lineage. Second, I will talk about stochastic simulation and the Gillespie method. Third, I will talk about the AE lineage simulation, and finishing it off with future direction. So here on the left, we see a cartoon model, and on the right, we see a histology model of the AE. The AE is primarily made up of 3 main cell types, being number 1, the secretory cell, number 2, the stem cell, and number 3, the ciliated cells. Now, the role of the secretory cell is to create mucus to trap any foreign antigen or particles that enter the body. The role of the stem cells is to heal any damage that may occur to any of the cells, and the role of the ciliated cells is to carry away any excess mucus or particles out of the body. So our goal is to create a computational model of the local dynamics of the AE lineage. the way we will accomplish this is by using stochastic simulation, which lends itself well to species with relatively low population numbers. Now here we see that it is made up of three main parts, being the stork matrix, which gives information on all the states that a species can transform into, the propensity function, which gives the probability that any given reaction will occur in the next infinitesimal time dt, and the chemical master equation, or CME for short, which gives the probability that each species will have a future population at a given future time. And here on the slide, we can see the general form of the CME, which the stochastic simulation was derived from. This form consists of coupled ODEs, which serves better for species with larger population sizes, which is outside of the scope of this model. So the way we will be accomplishing this simulation is by using the Gillespie method, more specifically the direct method, and then coding it all into MATLAB. Now, a quick overview of how it works is we start with a state X at a time t, then find two random variables and compute tau and j. From there, we'll substitute t with t plus tau and x with x plus the state change vector v sub j. Now, before we dive into the AE lineage, we will be needing to complete these example simulations presented on this table. Now here, highlighted in yellow, is the first example simulation, which is a simple degradation of reactant S. Now, in order to create the stork matrix, we put a -1 in the matrix to represent the degradation of reactant S. From there, the propensity function, denoted by A, is equal to the reaction constant C multiplied by X T sub I. So here we have the first three example simulations, which all share the same X and Y axes ranges to make comparisons more easily. So on the first example, we decided to run two trials on the same simulation to make sure that each trial has variations between themselves. As we can see on the graph, not only is S degrading over time, but the two simulations also have different results, which is what we wanted to see. Here for examples 2 and 3, we can see that reactants S, S1, and S2 are all degrading accordingly, and both of the products are increasing with time. For example 4, we can see that the reactant S is degrading much quicker, and this is due to the fact that if you can see the stork matrix, it'll have a -2 instead of a -1, and the product again is increasing with time. For example 5, we can see that reactants S1 and S2 are degrading with time accordingly. Product P is increasing with time, followed by decreasing with time due to it being the product of the first reaction and then a reactant in the second reaction. Now the final product, S3, is increasing in time, which is what we expected to see. With these simulations out of the way, we can finally move on to the AE simulation. On the left, you can see the local lineage control dynamics for the AE, which we got from the paper Near Equilibrium Calculus of Stem Cells in Application to the Airway Epithelium Lineage by Sun Zi et al. 2016. Now in this model, the stem cells are denoted by X, the secretory cells are denoted by Y, and the ciliated cells are denoted by Z. Furthermore, on this model, the solid arrows represent symmetric division, whereas the dashed lines represent asymmetric division. From this paper, we were also able to create the stork matrices, which you can see on the table to the right, and the propensity functions. Now, these propensity functions, as you will notice, depend on functions of X, Y, and Z. So we will have to program this accordingly into our simulation. Now, we decided to run 4 simulations for the AE lineage. And here we decided to run all of them with the same X and Y axes again to make sure that the comparisons are more easily drawn. Now, the stem cells will be denoted by the blue dashed lines, the ciliated cells by the yellow line, and the secretory cells by the orange line. Now, for homeostasis, we can see that not much changes between the beginning and end of the simulation, which is what we expected to see. For the only ciliated cells depletion, it normally wouldn't occur in nature, but we wanted to highlight how these cells would perform to damage. To begin the simulation, we started the ciliated cells at 10% of the homeostasis population. As you can see by this simulation, the ciliated cells do not make a full recovery by the end of the simulation. Now here we have the last two simulations we decided to run, being the depletion of both ciliated and secretory cells, and then the stem cell depletion, which again would not occur in nature. However, we wanted to see how the stem cells would react. Now here on the both ciliated and secretory cell simulation, we can see that the secretory cells make an immediate recovery to homeostasis levels, whereas the ciliated cells, by the end of the simulation, do not make a full recovery. Now, the stem cells depletion model, we can see that the stem cells increase immediately to homeostasis levels and remain there till the end of the simulation. Now, some future research which we will be conducting would be to compare this mixed model with the symmetric model, as you can see on the top left. And then furthermore, we will be adding to our simulation by adding spatial dynamics of the AE. For example, one area of the AE will have severe damage and another area will have little to no damage. Here are the papers that were used in reference to create the example and the AE lineage simulations. I would like to thank everyone who made this possible, and the floor is now open for questions.
Thank you very much, Wenamel. That was a great presentation. Really glad you actually were able to participate in a mathematics lab too, and take advantage with the biological system. Does anybody happen to have any questions for Manuel? Please feel free, you could type it into the webinar chat or raise your virtual hand. It looks like Mr. Andreas Perales has asked, what was your experience with running all of these simulations?
Well, at the start, it was pretty complicated because I didn't really know my way around MATLAB. And it was frustrating when it wasn't doing what I wanted it to do. But it was all just took a lot of patience to look over the code and make sure, you know, that everything was where it should be.
Okay. And Marissa is asking, did you already know how to code prior to your experience this summer?
Oh, prior to my experience this summer, I knew some C++, but not too complicated things. So there was some learning curve. However, I already had a head start on it.
All right. Does anybody happen to have any other questions for Manuel? Oh, Dr. Brown is making a comment. Our resident math major, despite being an epidemiologist, yeah, he is commenting. He had a great explanation, especially in terms of doing math and biology. It's a really great intersection. Might be something you want to be interested in, given the current climate of, yeah, given the current climate of today's world. And that was something that was new and interesting, is to discover that for myself, that epidemiology is the mathematical arm of health sciences. So something to even look into in the interdisciplinary aspects of other sciences. Well, thank you very much, Manuel. Great presentation. We will see you later on, and we'll move on to our next presenter.
Thank you.
Okay, so Mr. Joshua Wong, if you have the opportunity, we'll go ahead and start your presentation. Your PowerPoint is listed up and running, and you can take it away.
So can you hear me right now?
Yeah, we can hear you fine.
Great, okay. Hi, good morning. My name is Joshua Wong, and today I'll be presenting what I did for the RISE program, which is using density functional theory to calculate and predict the location of atomic germanium doping in silicon nanoclusters. So before we begin, let me give you guys some background on what my research is actually all about. Next slide, please. So in the modern era, we can actually see a trend towards miniaturization in electronics. So as this timeline indicates, there's actually a diminishing size of electronics from 1947 all the way up to now. And that process has actually also been accompanied by an increase in processing power. So now you might ask, well, where's this trend heading to? Where's this final goal? And to that, I have to say nanoclusters. So nanoclusters are basically larger molecules, right? And they've shown an incredible amount of complexity and customizability. So we can change conduction within these structures just by adding a substrate randomly across the nanocluster. And so this is interesting because this amount of complexity allows nanoclusters as a group to tackle a wide variety of problems that we see today and may see in the future. So next slide, please. I've actually selected a couple nanoclusters across the periodic table to further push this point home and make it easier for you guys to understand why nanoclusters and research into them is so important. So let's start off with the hexanuclear platinum cluster, which is on the very bottom. So research into this nanocluster has shown that there's a heightened stability because of its 6 cores. And so that's quite interesting because we can think about this in application in that the more cores that we have in electronics, the more stable it'll be, the more long life our electronic device might be as well. So the next nanocluster that I want to talk about is cobalt selenium cores and its families related to that. So research into cobalt selenium cores have shown that different variations off of it have different conduction values based on how well each nanocluster can access metal atoms within the core. And so basically what that kind of means is research has shine a light on just how we can manage conductance within nanoclusters by changing access towards these metal atoms. So the last nanocluster that I want to talk about is carborane, and that's on the top right. So in this image, we have 3 variations of carborane based on different connectivity. So on the left, we have orthocarborane, on the right, we have metacarborane, and on the bottom, we have paracarborane. And so basically what research has shown into these 3 variations of carborane is that each one of these has different conductance values. And that's quite interesting, right? Because the core nanocluster is all identical, it's just carborane. And so what this kind of implies is that it's not just the identity of nanoclusters that matters, but also its surrounding, like what its bond angle is and what kind of atoms it finds itself surrounded by. So to kind of demonstrate what I'm actually doing for RISE, I focused on one specific nanocluster, which is satellite adamantane. Next slide, please. So this is satellite adamantane. It's the fundamental unit of silicon. And the reason why research into this specific nanocluster is so important is because silicon plays an integral role in modern electronics, and satellite adamantane acts as a unit cell of bulk silicon. Thus, studying the electronic properties of this nanocluster shows promise in where we can go in the future of electronics. I then honed my study on the effects of germanium doping, which is basically replacing silicon atoms with germanium atoms. And this makes sense because germanium really likes to be substituted for silicon. So to give some more background on my personal research, basically what I did before the RISE program was I was actually physically in lab trying to synthesize satellite adamantane, and that's shown here in this synthetic scheme. So let me quickly break down what's happening. So on the left-hand side, we have a precursor, and then on the right-hand side, we actually have the nanocluster, which is satellite adamantane. And basically what's happening in this reaction is the bonds are forced to be reshuffled around to form a more thermodynamically favorable state, and that state would be the nanocluster satellite adamantane. So figure B is basically just a graphical representation of what's happening in figure A that's also in regards to the thermodynamic change of these structures. So unfortunately, research was actually moved online, so I had to shift the focus of my research a little bit. In that case, I shifted it to germanium-doped satellite adamantane synthesis. So as we can see in this slide, the synthetic schemes for germanium substituted precursors and the resulting isomers are quite similar to the previous slide, right? In fact, the only main difference is changing one silicon atom for germanium. So as we can see, I've changed that germanium to blue so it's easy for you guys to see what's going on. So because I've substituted germanium in for silicon in the precursor, there are now 3 isomers on the right-hand side based on different placements of the germanium atom. On the top, we have exocyclic germanium. On the bottom, we have endocyclic 1 germanium. And on the right-hand side, we have endocyclic 2 germanium. And so now you might ask, well, there are 3 isomers. Which one is the most likely result from this reaction? And that's the question that my research is trying to answer. Next slide. Thank you. So this is my computational project design. So as a reminder, the last and final step is to determine which isomer is the most likely isomer in this reaction. So the first step is to actually draw out these structures in Avogadro. Basically, Avogadro is this free software that you can download from the internet and allows you to model molecules. So you can draw a couple atoms in space and draw bonds in between them. So the next step is to perform molecular mechanics optimization on these models. And basically what that is it tries to find an idealized model for these molecules, right? And so let me break down that process for you. Basically, it treats each atom as spheres and it tries to get them to be as far apart from each other as possible, basically to maximize bond angles. So this process is quite interesting because by treating each atom as spheres, it kind of ignores the identity of these atoms. So it ignores like the amount of electrons, the amount of protons, neutrons, all that nice stuff that actually defines what an atom is. And because of that, it's actually quite inaccurate. And so to address this inaccuracy, I then run DFT optimizations in Gaussian. So Gaussian is another piece of software that you can download. And basically the way it works is it forces your setup to run multiple computations to answer a question that you pose it. So in this step, the question that I pose it would be, what's a more realistic model of this nanocluster that I just created in the previous two steps? And so basically the way it works is it'll accommodate for, it'll accommodate for electron densities, it'll accommodate for nuclei placements, and it'll accommodate for like the amount of protons and find a more realistic, more stable state that's actually based on the identities of each atom. And so now you might ask, well, isn't that the same thing as molecular mechanics optimization? And it kind of is, it's quite similar. It tries to find the most stable form, right? It's just more accurate and it actually counts for the identities. And so now your next question might be, well, because they're so similar, what's the point of molecular mechanics optimization? And so basically that point of molecular mechanics optimization is it provides a better starting point for DFT optimizations in Gaussian, since Gaussian is so heavy in terms of computation. So once we have this realistic model of our nanoclusters, we then perform frequency calculations again on Gaussian. And basically this time what Gaussian is looking for is related energies for these nanoclusters. So it also calculates for force constants, for example, which is the force related to moving around an atom within these structures. And so after this, the final and most important step is to extract an enthalpy value. And the reason why this step is so important is because by comparing enthalpy values with the isomers and referencing it back to the precursor, we can determine which isomer is the most likely isomer from this resulting reaction. So having said that, here are my results. So on the left-hand side, we have the germanium substituted precursors, and on the right-hand side, we have all three isomers from this reaction. So let me quickly draw your attention to the enthalpy value associated with the germanium precursor on the left. So as you can see, I set it to 0 kilocalories per mole, and that might strike you as a little strange. So the reason why I set this value to be 0 is because if I set the value for the precursor equal to 0, it would make it easier to reference the isomer values back to the precursor. So in short, it changes the reference frame to suit our specific needs. So moving on to the isomers, what Gaussian has shown me is that isocyclic germanium on the top has an enthalpy value of negative 32.9 kilocalories per mole. Endocyclic 1 germanium on the bottom has an enthalpy value of negative 34.2 kilocalories per mole. And on the right-hand side with endocyclic 2 germanium, I've shown an enthalpy value of negative 35.5 kilocalories per mole. So just by looking at this result, we can draw a few conclusions. So the most likely isomer to result from this reaction is endocyclic 2 germanium, and we can reach this conclusion by examining the deltas in enthalpy. More specifically, the delta in enthalpy between endocyclic 2 germanium and the precursor is the largest enthalpy by comparing that enthalpy value with the other two isomers. And another interesting note is that both the endocyclic species, so endocyclic 1 germanium and endocyclic 2 germanium, have relatively comparable values for enthalpy, right? It's negative 34.2 and negative 35.5. And so that's quite interesting because it seems like the endocyclic species are the more preferred species for synthesis as opposed to the exocyclic species. And so I guess to put it in other words, we could say that it wouldn't be unexpected to see a mixture of endocyclic 1 germanium and endocyclic 2 germanium with a preference towards endocyclic 2 germanium. And at this point in time, I'd also like to add a quick disclaimer for my calculations. So the calculations that I was running on my laptop weren't at the highest level of theory. So I had to cut a few corners for efficiency. And the reason behind that is because my laptop actually can't handle a high level of theory. And so as a result, my numbers may not be as accurate as I can get them to be. That's not to say that these numbers aren't accurate at all. It's just to say we can make them more accurate. So let me give you an example of the kind of corners that I've had to cut. Basically, with the level of theory I've been running, it kind of ignores some of the electronic interactions within these nanoclusters. For example, it doesn't really accommodate for the polarization of the orbitals. So looking forward to the future, I would like to work in person to chemically synthesize these nanoclusters, in essence, picking up where I left off from before the RISE program. I would also like to run calculations at a high level of theory to achieve a high level of accuracy. So before I open the floor to any questions, I would like to acknowledge the entire STU lab for all of their support throughout this entire process. I would also like to acknowledge the RISE coordinators for setting up this program despite all the challenges they've had to face along the way. And lastly, I would like to acknowledge Mr. Kenneth and Nancy Gray for providing the funding for my research. Thank you.
Thank you very much, Joshua. Great presentation. Does anybody happen to have any questions for Josh? Please feel free and type into the webinar chat or raise your virtual hand. We'd love to hear your voice as well. No, we'll give everybody a minute to contemplate if they'd like to ask you something. Sounds like very great job, and you did a lot of extensive work. Were you involved with this lab prior to the summer?
Yeah, I started like around midway through winter quarter, I want to say.
How was that transition, seeing as how you started in winter and then basically by the end of winter, we were being sent home?
Honestly, I think there were pros and cons for me personally. Like cons, obviously, I couldn't really hang around friends, right? But the pros is I got to see my family more. So I lived in the Bay, UCR RISE down in SoCal, and it was nice to be able to like see my family more on a day-to-day basis. So I would say the transition wasn't too harsh because I could see some of the silver lining in the process.
All right. Looks like you have a live question from Mr. Nathan Ortiz. Feel free to, we actually have unmuted you, Nathan, and you can ask.
Cool. Just want to point out, first of all, great job, by the way, presenting.
Thank you.
What expectations do you have in terms of like actually working in the lab and synthesizing the nanoparticles? Like, what do you expect from actually doing like the wet lab?
Oh, like what do you think will happen with these nanoclusters?
Yeah.
Okay, I see. So I was reading some research articles a while back on germanium substitution for silicon nanoclusters, right? And so there's this interesting article that was talking about solar cells with silicon-based solar cells, and then also substituting in germanium. And so basically what the results showed was germanium substituted solar cells actually last longer and they're more stable, but they also run at the same efficiency as just pure silicon solar cells. So to answer your question, I would expect these germanium substituted nanoclusters to be more stable and to have more longevity.
All right, thank you very much, Joshua, and thank you, Nate, for asking a question. We will be moving on to our next presenter. Congratulations. Okay, so next on our list is back again, Mr. Enrique Aguilar. Thank you for joining in. I see you're still at UCR. I seem to have moved, so I'm on the other side now.
Thank you, Nate, for the introduction again. And once again, good morning. And for those of you just joining, my name is Enrique CNAS, and I am a junior at the University of California, Riverside, and I am studying mechanical engineering. I transferred earlier this year from Glendale Community College, and for the past few months, I have been working with the OSCON Energy Lab. And a bit of background, I actually joined the project that was already in progress, so I kind of jumped in to help perform data analysis. And today I will be talking to you about using the galvanostatic intermittent titration technique to perform electrochemical characterizations of next generation lithium-ion batteries. Next slide. Oh, previous slide. I'm sorry about that. Okay, before diving in, however, I would like to talk a bit about the history of the lithium-ion battery. Sorry about that. The first commercially viable lithium-ion battery was created in the 1980s by the work of John B. Goodenough, Akira Yoshino, and M. Stanley Whittingham. For their work, they received the Nobel Prize of Chemistry in 1991. Batteries are typically composed of an anode, an electrolyte, and a cathode. The electrolyte, which is found between the anode and the cathode, is a medium which promotes the migration of lithium-ion batteries. These ions go back and forth from electrode to electrode, and this process results in the charging and discharging behavior of batteries. And as a result, of their ubiquitous applications, lithium-ion batteries have gone on to completely revolutionize the world. However, traditional batteries have reached their limits or are beginning to reach their limits. The continual advancements in modern technology have increased the demand on the performance of lithium-ion batteries, and thus they require higher energy density battery solutions. Conventional lithium-ion batteries are composed of metallic lithium oxides, such as cobalt oxide, manganese oxide, and aluminum oxide as the anode material. And carbon matrices are typically used as cathode material. And in recent years, lithium sulfur batteries have gained interest as being the next generation of lithium-ion batteries. And this has been primarily due to their higher theoretical energy density. And this is due to sulfur's lower atomic weight. Well, it's primarily due to sulfur's lower atomic weight and its ability to provide, on average, about 2 times more charge per mole. Additionally, the environmental impact of sulfur is relatively less detrimental when compared to the typical lithium-ion batteries. Also, there is an abundance of the elemental sulfur on Earth, so that's good. And in some applications, lithium sulfur batteries use a porous carbon sulfur matrix as the cathode material, and this matrix allows for sulfur to act as a conductor. On its own, sulfur is a bad conductor. So despite these promising characteristics, there are some issues with the lithium sulfur battery. One of the issues that was explored was the free migration of dissolved lithium polysulfides, also known as LiPs, between the electrodes. This effect is known as the shuttle effect. And although the dissolution of polysulfides aids in the use of active battery materials, there are some parasitic reactions that take place. Such negative effects are reduced Coulombic efficiency, a decreased utilization of the active material, and also an increased capacity fading. What all this means is that in practical applications, the lithium sulfur battery tends to rapidly degrade in performance as the charge and discharge cycles progress. In the OSCON lab, 3 lithium sulfur battery prototypes were produced, and these consisted of lithium metal anode and an acetylene black sulfur composite as the cathode material. And each of these 3 batteries, they consisted of varying electrolyte to sulfur content. So my primary responsibility this summer was to analyze the data sets that had been generated previously by the testing of the 3 lithium sulfur batteries. This data was thankfully gathered last year, so this testing was not affected by the pandemic and the shutdown and all of those things. So the battery testing method that was utilized is known as the galvanostatic intermittent titration technique, also known as GIT. And GIT applies a series of current pulses to the batteries that are being tested over a time period. And depending on whether the current is negative or positive, the battery will yield a different voltage profile. So above, you can see we have a voltage plotted over time, and the graphic may be a bit too small to see, but on the y-axis we have voltage, and on the x-axis we have time. And this time frame is several hours long. And as you can see, the voltage, the trend is positive. And if we go to the next slide. So here in the red, we have the current profile. And on the right-hand side, it's too small to see also, but we can see that current in red varies from 0 to about 0.1 amps over time. And if we look at the far left-hand side on the red line where it begins, we can see that this cycle begins with a charging current. And after a period of 10 minutes, the charging current is removed. So the current drops to 0. And as the test cycle begins a rest period of 10 minutes. So after 10 minutes, the current again is applied. and then the voltage changes. And then again, after 10 minutes, the charging current is removed, and then it goes back to a rest period. So this goes on and on until the predetermined test parameters are satisfied. If we go to the next slide. So I showed a charging cycle, but the similar process is followed for the discharge cycle. A negative current is applied and the voltage has a negative trend. And the testing of each battery goes on and on for hours and until the test is complete. Each charge and discharge cycle contains about 20,000 rows of data. And in the full test cycle of a battery, there are about 120,000 rows of data. And the battery characteristics that we were interested in were or are internal resistance, quasi-equilibrium state voltage, and chemical diffusion coefficients. And to analyze these characteristics, I developed Python programming scripts to aid in the analysis of these values. Let's see. Next slide. So here we see a small sample of some of the data we used in calculating the internal resistance profile of one charge cycle. On the left, you can see a snippet of the raw data generated. And on the right-hand side, you can see what the data looks like once it's been processed by my Python script. And on the left-hand side, if you look at the far left column, we can see that the values range from 0, then it's truncated, all the way up to about 20,000 rows of data. I was able to clean up all this data for one cycle to about 140 points of clean data on the right-hand side. Next slide. Again, here we see a sample of some of the data that we used on the left-hand side. And this time I am calculating the quasi-equilibrium state voltage as it varies over a charge cycle. On the left, you can see a snippet of the data again that's generated. And again, you can't see it here, but it goes from 0 to about 20,000 rows. And on the right-hand side, you can see what the data looks like once it's been processed by my Python script. And again, I was able to take about 20,000 rows of data and clean it up to just about 160 points of data. Next slide. Lastly, here we see some snippets of the work done to calculate the chemical diffusion coefficient and how it varied over time. And in short words, the chemical diffusion coefficient is the rate at which atoms diffuse. And with this expression shown above, we were able to calculate the value from those voltage profiles mentioned earlier. Next slide. So far, I've been able to take about 300,000 rows of data and consolidate it to about 3,000 rows of clean data, which are going to be graphed and analyzed next month. And in closing, throughout the month of October, I will continue to analyze lithium sulfur battery data with the scripts that I developed. After completing that, we will be working to compile graphs and try to reach conclusions as to what the data represents. And after completing this work, I will begin to work with COMSOL multiphysics software, which is a finite element analysis simulation tool to further investigate the chemical characterizations of these batteries. And last slide, I would like to thank Dr. Brandon Brown, Nhi Tran, Dr. Richard Cardullo, the National Science Foundation for providing the funding, and Dr. Cengiz Özkan, Tanner Zarin, and Vantage Advanced Technology also for providing funding for this project. And lastly, I would like to thank all the RISE mentors and particularly recognize Karina Morales and Isaiah Grant for their support. Thank you again. This concludes my presentation.
Thank you very much, Enrique. Great presentation. Does anybody have any questions for Enrique? And again, feel free, type in the chat or raise your virtual hand. I think we're kind of just right over time though. Oh, Dr. Brown does have a question for you, Enrique, and I will read it for you. How fulfilling was it to see processed data after cleaning all the thousands of rows of data? And to get to that point where lots of challenges and errors in programming overcome?
It was very satisfying. I spent, there were just a few scripts that were developed, but I spent all summer developing these scripts, reading through literature, trying to understand the mathematical foundations of these calculations that we were doing. and then having to translate those theoretical aspects into writing, solving the math by hand, and then having to transcribe, those ideas into a Python programming script. It was really challenging. And when I was able to see the cleaned up graphs and data, I was really happy.
And Radhika has a very quick question. Do lithium-ion batteries lose their charging capacity as they are used over time?
Yes, so that's the reason why your iPhone, you notice that it doesn't hold the charge as long, or why eventually, you need to replace your car batteries and things like that. I think I'm out of time. I'm happy to answer the question some other time. Thank you.
No worries. Thank you very much, Enrique. Great presentation. We'll be moving on to our next presenter, Ms. Imelda Funke. Your PowerPoint is loaded on the screen, and the floor is yours. Thank you.
Yeah. Thank you, Nee. Can you hear me?
Yeah, we hear you fine.
Okay. Hello there, everyone. Welcome to this presentation symposium. My name is Imelda Funke, and for the past 8 weeks, I've been working in Dr. Paloma's lab. In this lab, we performed computational study of the coronavirus main protease and the interaction with the protease with its N3 ligand. Next slide, Marissa. So for the overview of this presentation, I'll be covering first the background and the significance of this study, the goal of the project, materials and methods, results and discussion, the conclusion, references, and acknowledgement. Next slide, Marissa, please. So before I jump into the technical details, let me first give you a brief background and significance of this study. Coronavirus mutated from the SARS-CoV virus, which we had an outbreak back in 2003. COVID is a single-stranded positive sense RNA which infects its host cell, that is human and animals, and then inside the host cells, it undergoes transcription, translation to produce several copies of itself, so as to infect other neighboring cells. The main protease of COVID consists of an envelope protein called the E protein, which plays an important role in the shape of the virus, giving it its spherical shape. It also contains spike proteins that can be seen on the picture on your right hand. These spike proteins plays an important role in the entry of the virus into the host cell by attaching itself to the AC2 receptors found on the surface of the host cell. Coronavirus is infecting and killing the human race, which has led to a global pandemic and a shutdown of the global economy. The virus mode of transmission is through the pulmonary system, so it is important that we all wear masks to stop the spread of the virus. While we keep wearing our masks, it is also important that we defeat this virus together. But how can we defeat this virus other than wearing masks and practicing social distances? The only way out is to develop a therapeutic drug. In order to do so, there is a need to understand the coronavirus crystal and how it interacts with its ligand, which will be revealed in this study using camera and VMD. Next slide, Marissa, please. So the objectives of this study is to analyze the corona main protease crystal structure, to analyze the N3 ligand, and then to analyze the binding mechanism of the main protease to its ligand. And then we will finally propose a suitable mechanism for drug design as an antiviral treatment. Next slide, please. Okay. The materials and method used, we use software such as Camera that was downloaded from the UCSF Camera website. And we use that to analyze the primary, the secondary, the tertiary, and the quaternary structure of the main protease. Also, we utilize molecular dynamics, VMD, to analyze the active site of the main protease and how it interacts with the N3 ligand. We also utilize the code 6LU7 to download the crystals from the protein database website. Next slide, please. Okay, for the analysis of the primary structure, as you can see on the screen, it was observed by camera that the primary structure is made up of 360 amino acid residue consisting of mixed residues such as polar residue, hydrophobic residue, hydrophilic residues, and non-polar residues. These different residues are linked together by hydrogen bonds, forming two long polypeptide chains. Next slide. For the analysis of the secondary and tertiary structure and quaternary structure, it was seen and demonstrated by the picture on your right hand that the crystal constitutes of 3 domains, which are 7 left-handed alpha helix color-coded as brown, 10 beta-plated sheets color-coded as purple, some loop regions color-coded as blue, and also the M3 ligand attached to the main protease color-coded as green sticks. Next slide. So when analyzing the active site of the main protease by camera and VMT, it will reveal that the active site is embedded within the beta-plated sheet domain of the main protease, and the active site constitutes of a catalytic diad made up of histidine and glutamate. There were also some surrounding amino acid residues observed around the active site, which interact with the diads through hydrogen bonds and pi bonds. As seen on the picture at your right hand, hydrophobic residues were coded as white, hydrophilic residues were coded as green, positive amino residues were color-coded as blue, and the negative amino acid residues were color-coded as red. Also, you can see that the active sites interact with the ligand, as you can see on the picture on the screen. Next slide, Marissa. The next thing was to analyze the mobility at the active site. We used the root mean square deviation to calculate the mobility of the active site and the ligand so as to determine the stability. RMSD value were calculated where the y-axis was in Armstrong and the x-axis in frame number. Lower RMSD indicates lower mobility and high stability, while high RMSD indicates high mobility and hence instability. As seen on the screen, which shows that the RMSD for just the active site with no ligand attached, low mobility was observed of about 2 Armstrong. Next slide, please. When the RMSD of the center of the active site was analyzed, low RMSD value was obtained due to the presence of 3 hydrogen bonds, which were located at the center of the active site, creating more stability. But when the RMSD of the ligand was analyzed, the end of the ligand shows high RMSD value for greater mobility, showing more spikes, as you can see on the screen. Next slide, please. The RMSD at the end of the ligand was greater due to the presence of an aromatic ring residues, creating more weight and instability at the end of the ligand, thereby reducing the strength at which the ligand interacts with the active site. Next slide. So in conclusion, it was seen that there were 3 hydrogen bonds present at the center of the active site were most important. It reduces less stability by RMSD value. It kept the ligand bounded to the active site. There was also an aromatic residue present at the end of the ligand, creating instability at the active site due to its large size. So to solve this problem, we need to alter the aromatic residue with a smaller amino acid residue so as to create more stability for the ligand to bind tightly to its active site. Hence, to increase affinity of the ligand to its active site, it is important to carry out a single point mutation at the position of the aromatic acid residue found on the ligand attached to the active site for therapeutic treatment to work. Next slide, please. These are my references. Next slide, Marissa. Thank you for listening to my presentation. And before I go ahead to take any questions and concerns, I would love to acknowledge and thank the following people for their help and support throughout this wonderful virtual research experience. Dr. Paloma, my PI, Brandon Michel, who is the TA in the lab that helped me to design all this presentation and helped me throughout this research process. The RISE funding donors, the RISE coordinator, Dr. Brandon Brown, Nitran, and all my peer mentors for that kind assistance and motivation. Thank you very much. Any question?
Noted his seal of approval for you as well. That was perfectly on time. Does anybody have any questions for Imelda? Please feel free, type it into the chat, or you can raise your hand. We'd love to. Oh, if you're PI. What were some of the challenges?
Sorry? Oh my God. I can hardly hear. Huh?
Can you hear me now?
Yeah, I can hear you, sorry. Yeah.
Some of the challenges that you encountered since you had been working with your professor prior to the pandemic or in the transition coming into the summer?
The challenges was just to get hold of my professor sometimes, my PI, it was quite challenging. And also to walk through these different softwares to get those beautiful pictures that I put on the slides. I came from a background where I have zero knowledge on coding, so I had to do all of this by myself. And I really, really thank this research experience that it has taught me to explore those parts of me that I didn't know I had inside of me. So thank you very much.
To our next and last presentation of this morning session. We can
Good to go. Okay. Hello, everyone. My name is Abigail Lusunde, as Nia already said. And throughout these 8 weeks, I have been working with Dr. Terry Nottingham, who is my PI, and Arzu Ulu, who's assistant project scientist. And we worked on nutrition and occupational exposure in farmland or with farm workers and the impact of occupational exposure. Next slide. So occupational exposure can be defined in my own terms after reading a lot of literature as different exposures or exposums that farm workers are exposed to while working that in turn leads to various respiratory disease and or health problems. Let's be real, farms are very, very dusty. And this dust are what these farm workers breathe in every day when they go to work. And this dust leads to irritation and occupational induced pulmonary disease. This occupational exposure comes in form of dust, vapor, fumes, and bioaerosols. Next slide. This topic is prevalent today as we know that there has been a lot of ashes due to the wild California fire, and these ashes do not do good to the body or its respiratory system. And so the impact of occupational exposure on these farm workers is that it contributes to the inflammation that is part of the progression of lung diseases such as non-allergic asthma, chronic bronchitis, and chronic obstructive pulmonary disease, which is also known as COPD. Occupational exposure has a big medical and economic burden on the society, as 10 to 25% of pulmonary disease is due to occupational exposure. And on the right, there is a picture showing what all this exposure does to the lungs. Next. Also, not only are these farm workers at risk of developing occupational exposure, occupational pulmonary diseases. Also, those living around the area and their family members are also at risk of developing these pulmonary diseases. Next slide. What did we find? All right, through all the research done, we found out that nutrition is associated with lung function and could potentially be important in shaping an individual's disease risk. We also found out that increased intakes of certain nutrients, especially those with antioxidant and anti-inflammatory properties, were positively associated with pulmonary function. Next. We also found out that micronutrients such as omega-3 polyunsaturated acids, vitamin D, and zinc all have potential in the protection against the adverse respiratory effects. And we found out that proper preventive equipment were not provided for these workers. Yes, some equipment were provided for them, but due to the nature of their work, some of these equipment were either heavy, uncomfortable, or hard to breathe in with. Next. The role of these micronutrients in occupational exposure, which is omega-3 fatty acids, vitamin D, and zinc. Omega-3 fatty acids gives rise to anti-inflammatory specialized pro-resolving lipid mediators that protect endogenous resolution of inflammation and protects against inflammatory stimuli. Vitamin D regulates the expressions of genes in brachial smooth muscle cells, includes increased secretion of antimicrobial peptide cathelicidin, and these cellular effects are important for host responses against infection and the development of allergic lung diseases like asthma. Next. Lastly, we looked at zinc, and zinc is an essential trace element required for maintaining intestinal cells, bone growth, and immune function and defense against pathogens. Zinc also protects the health and integrity of the respiratory cells during lung inflammation or injuries. The methods by which we figured out that these micronutrients show positive effect on occupational induced pulmonary diseases or occupational exposure, Dr. Nordingen collected biological samples from animals that had been exposed to extracts of dust or particulates that are applicable to exposures of farm workers. And she found out that omega-3 fatty acid, the micronutrient, helped these animals to get better or less sick. Reading other literatures about occupational exposure also showed that vitamin D and zinc also had positive effects on occupational exposure following the same procedures or methods. Next slide. Future directions that needs to be put in place to better the health of farm workers are ensuring the availability of proper preventive equipment or PPE for these workers. And on the right, there's a picture of the proper equipment that should be provided and can be provided for these workers. Training personnel in infection control and procedures. Conducting nutrition in agricultural populations. Next slide. Also, regular workplace inspection and cleaning out the factory, making sure that the air in the environment is breathable and it's clean. Develop plans with measures to protect workers during emergencies or non-routine activities. Because while reading some literatures, we also found out that most of these workers, after these diseases become super severe, they're laid off and no further health services being provided for them. And of course, nutritional recommendations should be made available, should be made at disposal of these workers and not scarce or not available for these workers. And then the next slide. And in conclusion, our findings indicate an important role for the protective effect of certain nutrients against inflammation associated with occupational exposure. Yes, occupational exposures continues to be a major problem among agricultural workers, but if the right measures or guidelines are taken in place, it may help mitigate the negative impact of occupational inhalant exposure. And a side note, our paper has been submitted to a journal named Current Allergy and Asthma for publishing. And the title of this paper is Nutritional Factors in Occupational Lung Diseases. And below, you can also see the names of the authors that helped in contributing to write this paper. And these are my references, and that concludes my presentation. And I would like to thank NIH for funding the B2B program, Dr. Isaac and Dr. Ford, who are the former PIs for this grant. I would also like to thank Dr. Scott Herrick, who was the primary contact at RCC, Dr. Nordigan, who allowed me to work in her lab, Arzu Ulu, who also guided me this past weeks, helping me with my project, helping me with also my symposium, Nitran, Marissa, all the peer mentors, and everyone that contributed in making this program a fun and eventful one while being online. Now I open the floor for questions.
I will second Jalen's comment there. Amazing presentation. Thank you very much, Abigail. Does anybody have any questions? And that's a great way to close out this particular first session. A lot of names I haven't seen. Dr. Herrick, a very good friend, as well as a collaborator. He was a UCR alum and got his PhD under Dr. Richard Cardullo, who is in attendance today. And if anybody has questions, feel free. You can type it into the chat, or you can raise your virtual hand and we can unmute you so you can ask Abigail live. So I know this was your first experience with research, and you're still currently going or taking courses at RCC, is that correct?
Right.
All right, that's obviously been a challenge. What what have you taken or from this particular experience over the summer, and what are your plans for the future?
From this summer, I've actually taken to that. If you don't actually go out of your comfort zone, you don't actually know what's going on. And if you don't actually read about stuff, you wouldn't actually know that certain things are happening. Because before this, working in the lab, I didn't know about what occupational exposure meant and what is the harmful effect of it. And so I basically learned that you have to go beyond and above to just educate yourself and become more real with the environment around you.
All right, thank you very much. So Marissa does have a question for you. How has this research changed your view of farm workers?
Right now, it's changed my view a lot. These farm workers are working a lot, and most of the time they're taken for granted because we don't actually see what they're doing. We only see the product of what they have done already. And so it's really changed my mind in their view.
Okay. Thank you very much. Again, amazing presentation. Great job. We will now commence the beginning of our 5-minute break. Right now it's 10:20. We will resume back at 10:25 with additional oral presentations, and then we'll jump into the second half, some additional poster presentations. So everyone please get up, have a restroom break, stretch, get out of that seat, try to grab a quick snack. We will see you in 5 minutes. Thank you. welcome back, everybody. That was, as usual, a very, very quick 5 minutes, but I was able to run and grab a little bit of coffee to keep me up for the next couple hours. But with the way these presentations are going, I don't need too much caffeine. We have our next presenter, Ms. Noor Hamzi. Thank you very much for turning on your camera. Your PowerPoint is loaded on the screen now. And floor is yours. You can take it away. Thank you.
Thank you. I am part of Dr. Adam Godzik Lab, who is a computational scientist working on a project conducted by the mentioned authors that coalesces around the role of smoking effects on tumor immune microenvironment. Next slide, please. Can you hear me? There are a great number of research articles that supported the smoking affects the gene expression profiles and DNA mutation due to its genotoxicity, but it also has major effects on the tumor immune, on the host tumor immune system. American Cancer Society estimated that roughly 700,000 people would die of cancer, and over a quarter of that number is attributed to smoking cigarettes. The reason behind this, the reason behind this implication is that tobacco contains carcinogenic components that can damage the DNA, increasing the risk cancer initiation and progression. The study used genomic cancer atlas data to analyze the smoking effects on gene expression profile, but also all of these samples from the genomic atlas data focused on the smoking effects on gene expression profile, but we expanded this focus to study the impact of tobacco on immune system. Next slide, please. Next slide, please. Comparative analysis of tobacco smoking induced changes in populations of tumor infiltrating cells were performed in 10 TCGA cancer patients. Tumor infiltrating cells are pivotal elements in both innate immunity and adaptive immunity, and they can recognize and kill cancer cells. They're represented on the left figure on the horizontal line, and this figure shows the most affected immune cells, tumor infiltrating immune cells for every cancer type for both genders, how their quantity differentiates in smokers compared to never smokers. This shows a significant observation differences in the quantity or the populations of immune cell types for every cancer patient for females and males. Next slide, please. The figure on the right is complementary to the figure on the left. The figure on the left, as mentioned earlier, only depicts the different ratios or in populations of the immune cell type for every cancer type. However, the figure on the right correlates these results with survival probability. Let's take, for example, graph B. Graph B, the accentuated red curve, represents the high ratio of activated to resting natural killer cells that are part of the innate immunity in current smokers. And from the graph, we can infer that there's a higher survival probability in current smokers with a high ratio of activated to resting natural killer cells compared to the faded curve that represents the lower ratio. This result is also supported by graph A, where it depicts the ratio in non-smokers as our control group. For example, the blue curve in graph A, which is the very first curve, represents the high ratio of activated to resting natural killer cells. And those group, the non-smokers group, experienced the highest survival probability because it ranged from 0.75 to from 1 to 0.75. So the trend here is positive correlation. The higher the ratio of activated to resting natural killer cells, the higher the survival probability. This also suggests that in smoking, this ratio is susceptible to reduction. And The reduced ratio of activated to resting natural killer cells could be one of the main reasons why current smokers experience a severe low survival probability compared to current smokers and never smokers. This result is also consistent with figure 1 on the left, where if you looked activating to resting natural killer cells experienced more green color code, and green color code represents a decrease in quantity of that particular immune cells, and the red codes for an increase in quantity. Next slide, please. This is another observation in the immune cell population, where plasma cells to memory B cells dramatically increased in smokers, and that's for both sexes. Also, the figure on the left, indicated by mean decrease Gini, recorded that the most affected classes of tumor infiltrating cells are the plasma cells, and that's for both genders. Next slide, please. Now, in terms of smoking effects on the immunity at the gene expression level, GPR15 was found to be the only significantly differentially expressed gene between smokers and never smokers, and this result is consistent with both genders. Now, the physiological role of this gene remains elusive, but it is known to be prominently located in T cells. The smoking-related elevation in both the GPR15 expression and the increased ratio of plasma cells to memory B cells suggests that immune cells can respond to the same stimulus. However, it could also suggest that the increased ratio of GPR15 T cells and plasma cells to memory B cells are negatively correlated with current smokers compared to never smokers or former smokers. This is another strong evidence on the role of smoking to the immune system. Yes, tobacco does affect gene expression profile, but not a lot of authors considered that smoking differentially affects the expression of genes based on the immune cell type. For example, the gene MPDU1 was expressed at a much higher rate in smokers in CD4 T cells. Whereas when it was located in CD8 T cells, its expression results showed actually opposite effects. Another example of the same gene being located in different immune cell type is KLRC1 gene, and it was expressed at a much higher rate in smokers compared to never smokers for CD8 T cells. Next slide, please. In summary, tumor does not only damages the DNA leading to mutation due to its carcinogenic features, but it also affects the population ratios of the immune cells. Interestingly, minor shifts in the populations of tumor infiltrating cells that are not statistically significant by themselves can yet lead to a substantial decrease in the survival rate of cancer patients who are active smokers. For example, CD4 memory T cells, when they are expressed at a much higher rate, it showed low probability. However, when we look at the first data, figure 1, this type of cell did not show a significant decrease or increase in its quantity, but yet it still led to a significant low survival probability, despite its minor shifts in its statistical difference in its population. Next slide, please. There are some gender-specific differences in immune cells populations, and one of the most general observations is that the changes in immune cells populations in women are more pronounced in men. Women's immune system responds differently than in men. Next slide, please. Female smokers pay a much higher price in terms of smoking-related diseases and deaths. For example, in the middle graph of figure A, women showed that they experience a severely low survival probability when plasma cells are being produced at a much higher rate compared to male current smokers, which is at the graph that is directly below that. Next slide, please. For future references, we want to understand better why women respond differently to smoking than men. The three major methods that we use are three databases: TCGA cohort, the RNA sequence data, and PanAtlas publication page. The TCGA provided over 10 samples, and we used 2,724 samples were investigated, and these samples were divided into three categories based on smoking status: former smokers, current smokers, and never smokers, to analyze the smoking effects of on the gene expression level and on the immune cells populations. The RNA sequence data was used to analyze the smoking related effects under gene expression level. The R package of survival and TCGA ABO links and log rank calculations was used to calculate the survival analysis that is correlated with different immune cells population between smokers and never smokers. And p-values was calculated for every survival analytical graph, and it showed in agreement with the original claims. Next slide. I would like to acknowledge Dr. Byron for Dr. Herrick for offering me the opportunity to be part of RISE program through B2B program that is funded by National Institution of General Medical Sciences and NIH. I would also want to give special thanks to the lab mates that were part of the research paper and my PI for guiding me into analytical thinking and explaining the experimental approaches.
Thank you very much, Noor. All right, that was a great presentation. And fortunately, we have run over a little bit of time. If anybody happens to have questions, you can try to chat or you can send a question through the chat. And I think we may have time. Dr. Nugent did ask you, would you expect secondhand smoke exposure to show similar impacts on gene expression?
That's a really good question. We did not study the differential concentrations of tobacco on immune system because I think the secondhand smoke would have a lower concentration of carcinogenic components that can affect the immunity, but we haven't studied that. I think the key here would lie on the concentration of carcinogenic components that are in the tobacco, such as nicotine.
All right, thank you very much. All right, we will be moving on to our next presenter, Mr. Taylor Oliver. Thank you very much. Nice background. Your presentation is already loaded, so I'll let you take it away. Thank you.
Thank you. Good morning, everyone. I'm Taylor Oliver, and for the past few weeks, I've been learning about the parasite Toxoplasma gondii in Dr. Emma Wilson's lab. Unfortunately, I haven't been able to complete research of my own, but I have plenty to share with you today. Let's begin. T. gondii is an obligate intracellular protozoan parasite, which means it predominantly lives in cells and can invade any nucleated cell. Toxoplasma is one of the most successful pathogens on the planet, with prevalence rates in Europe and the USA between 15 and 80%. No mammal or bird population has yet been found that does not harbor Toxoplasma. Upon infection, an acute inflammatory response occurs. The response is controlled and results in a chronic phase of infection wherein the parasite is contained predominantly as a slow replicating form in the central nervous system. Many individuals are asymptomatic due to a robust immune response, and most notably, symptoms may be flu-like and or swelling of the cervical lymph nodes. Lacking such an immune response, parasite reactivation may occur, leading to toxoplasmic encephalitis, a potentially fatal disease if left untreated. An anomaly of T. gondii is that while infection rates are high, instances of toxoplasmic encephalitis are rare, though on occurrence it is severe. Next slide, please. Retinal toxoplasmosis is the uncontrolled replication of the parasite in the retina and is one of the leading causes of blindness. Congenitally acquired toxoplasma infections remain one of the largest causes of fetal abnormalities, including hydrocephalus, intracranial calcification, and mental retardation. In the situation where a compromised immune system is present, such as HIV and AIDS, patients can suffer neurological conditions due to the reactivation of the parasite from cysts. It is now recognized that all genetic or acquired immune compromised patients are susceptible and toxoplasmosis is reported following organ transplant or chemotherapy. Further study of T. gondii can lead to not only a potential treatment for the parasite itself, but also a better understanding of the immune system and its responses, and a number of neurological disorders such as autism spectrum disorder, bipolar disorder, schizophrenia, and Huntington's disease, Alzheimer's disease, amyotrophic lateral sclerosis, and multiple sclerosis due to their relationships with neurotransmitters. Next slide, please. Actually, I think that's the slide I'm supposed to be on right now, the previous slide. So to begin to understand the parasite, we will start with its life cycle. The definitive host of T. gondii are felids or felines, the domestic cat being a prime example. It is here where sexual reproduction of the parasite is able to take place. Because felines are the definitive host, the infection of any other hosts are seen as intermediates. Following infection of an intermediate host, cysts rupture and sporozoites from oocysts or bradiozoites from tissue cysts are released into the interior of the gut. Parasites invade the cells of the small intestine, then convert to fast-replicating tachyozoites. From there, they replicate and spread throughout the host body and tissues, reconverting the bradiozoites and forming tissue cysts. After infecting a feline, oocysts are produced in the gut, and for about 2 weeks, the cat will shed oocysts in its feces, leading to environmental contamination and spread. Next slide, please. So Toxoplasma gondii infects approximately a third of the world's population, making it one of the most prominent foodborne pathogens. Ingestion of T. gondii by any mammal other than a cat leads to the development of tissue cysts. These cysts are formed in muscle, heart, liver, and brain tissues, and they remain in the tissue for the lifetime of the host. Humans can become infected with T. gondii from ingesting unwashed fruits and vegetables that have been contaminated with oocysts. Contact with contaminated feces can also lead to an infection, as well as contaminated water sources. Due to environmental contamination by feces, grazing animals can become infected, providing another pathway for infection in humans through the consumption of undercooked meat. As intermediate hosts, human infections represent a dead end for the parasite, leading to a lifelong infection, but for most individuals, the infection goes unnoticed. Next slide, please. How does it start? First, T. gondii enters the interior of the gut. The parasite then proceeds to spread throughout the host tissues, sometimes hijacking the host's own immune cells, continuing to reproduce until the host's immune response is sufficient to control the infection. Infestation of the parasite occurs in the host tissues, predominantly in the brain, but also skeletal muscle as tachyozoites convert to bradiozoites and form tissue cysts. To infect the brain, the parasite must cross the blood-brain barrier, typically not an easy feat, after which it can begin to infect the resident cells of the brain like neurons. The method of crossing the blood-brain barrier is subject of study, but potential pathways have been identified, such as transcytosis and parasitosis. Transcytosis is a type of transcellular transport where vesicles move across the interior of the cell from one end to another. Parasitosis involves crossing at junctions between cells, typically where cells communicate with adjacent cells. And then there's the Trojan horse method, which usually uses infected immune cells to cross the blood-brain barrier, such as macrophages and dendritic cells. Next slide, please. So what happens to the central nervous system cells, resident cells following infection? The neurotransmitter glutamate is arguably the most important neurotransmitter in the brain, and unregulated levels can cause nerve damage. Therefore, central nervous system glutamate is strictly controlled. Glutamate is released at presynaptic sites during synaptic transmission. The extracellular glutamate is regulated by action-specific transporters present on astrocytes. The main astrocytic glutamate transporter in the forebrain is GLT1, also known as glutamate transporter 1. After crossing the blood-brain barrier, the parasite forms tissue cysts within neurons. Its presence in the brain has been associated with abnormalities in the levels of the neurotransmitter glutamate, as well as glutamate's regulation by astrocytes, caused by the morphological and physiological changes in both astrocytes and neurons, such as swelling. One of the major roles of astrocytes is to remove extracellular glutamate to prevent nerve damage. These changes lead to poor regulation of glutamate via downregulation of GLT1, causing increased extracellular glutamate levels, interfering with the neuron's ability to communicate properly. The presence of the parasite and infection-induced inflammation can lead to a combination of increased excitatory and decreased inhibitory neurotransmission, causing increased susceptibility to seizures and other potential neurological disorders. Next slide, please. Looking at the immune response, while the brain is an immune privileged area of the body, an immune response does occur. The most dominant immune cells in the brain are microglia, which make up 80% of brain immune cells. Other immune cells identified in the brain include myeloid cells, monocytes and macrophages, dendritic cells, T cells, B cells, and natural killer cells. Following challenge with T. gondii, monocytes, neutrophils, and dendritic cells are recruited to the site of infection. All of these cell types have been implicated in resistance to T. gondii. One of the most critical functions of the innate immune response to T. gondii is the ability to sense the pathogen and produce the cytokine interleukin-12, which stimulates natural killer cells and T cells to produce the cytokine interferon gamma. Interferon gamma is the major mediator of resistance to T. gondii and promotes multiple intracellular mechanisms to kill the parasite and inhibit its replication. This helper T cell immune response, defined by the production of interleukin-12 and interferon gamma, is characteristic of infection with many intracellular pathogens. In the brain, microglia as well as infiltrating T cells are the primary sources of interferon gamma. Next slide, please. Continuing with the immune response, the innate production of interleukin-12 during toxoplasmosis requires that the parasite first be sensed by the host. Innate immune receptors called toll-like receptors appear to have a role in this process. Toll-like receptors are a class of proteins that play a key role in the innate immune system response. They are single-pass membrane spanning receptors usually expressed on sentinel cells such as macrophages and dendritic cells that recognize structurally conserved molecules derived from microbes. Once these microbes have breached physical barriers such as the skin or the intestinal tract mucosa, they are recognized by TLRs, which activate immune cell responses. Astrocytes also play a role in regulating the infection of T. gondii, while having the important job of regulating central nervous system glutamate by adjusting uptake, release, and synthesis into glutamine. These cells also play an active role in controlling toxoplasma infection with the ability to upregulate pro-inflammatory cytokines, secrete chemoattractants, and internally kill parasites via IgTP. Next slide, please. The balance of this immune response is key. Balancing both the pro-inflammatory response and the anti-inflammatory responses is vital for the health of the host. The brain is enclosed within the rigid casing of the skull, making the immune cell recruitment and swelling normally associated with immune responses a potentially fatal outcome. As with acute infections and most other type 1 responses, production of interferon gamma and other pro-inflammatory cytokines must be regulated by anti-inflammatory cytokines, including interleukin-10 and 27, because continued recruitment of highly activated immune cells to the brain leads to encephalitis independent of parasite burden. Helper 2 T cells help to counteract the response to helper 1 T cells to prevent damage by inflammation via interleukin-10. Helper 2 T cells also produce interleukins like interleukin 4, 5, and 13. There is normally low expression of antigen presenting molecules, and the blood-brain barrier limits immune cells and antibody infiltration. The prevalence of toxoplasma and the requirement for ongoing immunity to the parasite in the brain suggest a highly controlled immune response that is robust enough to contain the parasite, yet limited to prevent clinical symptoms. The dynamics of this response are only beginning to be unraveled. In conclusion, with its capability to invade any cell, it is no wonder the parasite is so widespread. And due to its ability to suppress its host immune system, it is an ideal system for parasite immunologists and the gold standard for the study of type 1 immune responses due to mice being a natural host. The infection caused by T. gondii is a rare example of a well-balanced and protective immune response to the brain. By better understanding the interaction between the parasite and the host immune response, more can be learned of controlled immunity in the central nervous system and insight gained into immune responses and the capability to affect improper responses, as well as further insight into the central nervous system itself. You can move on to acknowledgements. I'd like to thank everyone who made this presentation possible. RISE Summer Research in Science and Engineering, the Bridges to Baccalaureate Program, the National Institutes of Health, and National Institute of General Medical Sciences. Also, a big thank you to Dr. Emma Wilson for allowing me to participate in her lab. And if there's any questions, I can take them now.
Thank you very much, Taylor. Great job. If anybody happens to have questions, feel free to type in the chat We did go over a little bit of time, which is okay. And I'll allow them to type them in, but what we can do, oh, Enrique is not sure how he feels about food now. Well, still fine to eat, just make sure you don't have too many cats nearby. That's probably the bigger takeaway if you're going to be sharing food with any feline. We will be moving on to our next presenter. Thank you very much, Taylor. Great presentation.
Thank you.
All right. So we have Ms. Natalie. Thank you very much for turning on your camera. Your PowerPoint is up and running, so the floor is yours.
Hi, my name is Natalie Masai. I'm a biology major going into my 3rd year, and for the past 8 weeks, I've had the opportunity of working in Dr. David Lowe's lab, and we have been looking at developing a method of quantifying data from our histopathology analysis of ileitis. Next slide, please. So we are looking at a TNF-DARE mouse model of intestinal inflammation. This model has a systemic overexpression of TNF-alpha leading to ileitis. Groups of mice are being treated with anti-TNF-alpha drug treatments in order to determine their effectiveness. From there, we develop H&E images to study the tissue. We are looking for ways to quantify data from these images. We believe that since healthy villi tend to be longer and thinner, that the diseased villi will be shorter and wider. Next slide, please. Thank you. Just for reference, this is what a villi looks like. The inner tissue is called the lamina propria, and this is where all the cells infiltrate when there's disease, leading to our belief that as a result, the villi will be shorter and wider. The area under and next to the villi is called the crypt. Next slide, please. There are 2 different kinds of inflammatory bowel disease, which is an autoimmune disease where there is an inflammation of the intestinal tract. The 2 kinds are Crohn's disease and ulcerative colitis. Our focus is on Crohn's disease, which mainly affects the terminal ileum. Now, like most other autoimmune diseases, it appears spontaneously and the exact cause is unknown. Next slide, please. We are using a TNF-DARE mouse model to look at the disease. This mouse model has a genetic mutation where there is a deletion of AU-rich elements, thus resulting in a systemic overexpression of TNF-alpha. TNF-alpha is a part of the innate immune response. It is a pro-inflammatory cytokine that activates endothelial cells. So since it is a systemic overexpression, it can be seen in the mouse's tissue throughout its body and not just in the gastrointestinal tract where there's disease. Its overexpression leads to an ileitis, which is an inflammation of the ileum, and this ileitis has features very similar to those of Crohn's, allowing it to be a good model to study. We're generally looking at 4 to 8 and 10-week-old mice. Next slide, please. We are using histology to assess the disease progression due to the recruitment of inflammatory cells into the lamina propria. The idea is that the histology is altered as the disease worsens. The lab is also getting gene expression data to correlate inflammatory gene induction with histopathology changes. Next slide, please. In the lab, H&E images are developed of the intestinal tissue of the mouse. The mice are euthanized and the intestine is removed. The terminal ileum is approximately 6 centimeters long and about 2 centimeters are removed for RNA extraction. After that, several small sections of approximately 1 to 2 millimeters are cut. These have been taken out both longitudinally, which can be seen in the image to the right of the slide, and in a donut form, which can be seen in the image to the bottom. The sections are placed in a fixative and then a square mold with OCT and then flash frozen. Sections of approximately 15 micrometer thickness are cut from this frozen block using a cryostat, and then they are placed on electrostatically charged slides in order to prevent any fragmentation or the tissue moving. They're stained with hemotoxylin and eosin. Hemotoxylin is a positively charged staining solution that appears dark purple, and it allows us to see the nuclei of the epithelial cells, while eosin is a negatively charged staining solution, and it appears pink and stains the cytoplasm, basement membrane, and muscularis layer of the membrane. Using a bright light microscope, the images are taken at a 20x objective, and then they're transferred to Photoshop, where they're stitched together in a way that still maintains their resolution. And then the images are coded with a naming convention that I do not know, and they are sent to me. So the images are coded so that I don't know whether the image I'm receiving is of a healthy tissue, diseased tissue, the age of the mouse, or any other identifying factors. We've chosen to do these blind measurements so that we can have an objective view on the data being collected and be able to draw accurate conclusions from them, as well as be able to develop an unbiased quantitative analysis method, as opposed to assuming the data means something on account of already knowing what is diseased and what is healthy, or developing a biased method. Something that also needs to be kept in mind when we're taking these measurements is that Crohn's disease is a patchy disease, and this means that while a section of the tissue may appear diseased, another section may appear healthy. Next slide, please. So as I stated before, we're currently trying to determine whether or not, since healthy villi are long and thin, if the diseased villi would be short and wide as a result of all the cells infiltrating the lamina propria. So for our first method, we took a basic length and width measurement using image J, and we use image J for all our measurements. The length was taken from the top of the villus to the bottom of the crypt at the center of the villi, and the width was taken at the halfway point of the length value. This point is given to us by the line tool being used on image J. The yellow lines on the image show which villi were measured and where. We then took a ratio of the length over the width, and we didn't use a scale bar and stuck to using a ratio to eliminate having to worry about a consistent scale bar across all the measurements. Our restrictions for choosing the villi were pretty lenient in a sense that as long as it wasn't slanted to the extent where we can't reach the top of the villi, then it can be measured and the villi can't be disconnected from the rest of the tissue. But we felt that these restrictions were a little too lenient and didn't provide for accurate measurements, because as you can see, there are villi being measured where the top is fragmented off, which wouldn't be an accurate length measurement. Next slide, please. So this is where method 2 came in. This time our restrictions were stricter in the sense that the villi cannot be slanted or fragmented whatsoever. The idea is that a line can be drawn from the top of the villi to the bottom of the crypt without leaving the villi at any time or accounting for any gaps in the length. The villi with arrows pointing at them were the ones included in the original measurement, but as you can see, they're not in the best condition. The ones with the length and width lines on them are the ones measured with our new restrictions that we're going to use for the rest of the measurements. So the length and width were taken in the exact same way, and once again, there was no scale bar, and we took a ratio for the length over the width. However, we were noticing that there were times that the halfway point for the width was below the villi, and this can be seen with the first measured villi to my left. We felt that wasn't an accurate measurement of the width, since the line was not being drawn from one side of the villi to the other, but rather from what we approximate to be one side of the villi to the other. So from there, we developed method 3. Next slide, please. In method 3, the length was taken the same once again. However, after taking the length measurement, we manually calculated a quarter of the length value, and that is where we took our width. This guaranteed that the width measurement was taken inside the villi at all times. However, it can be difficult to find the exact value using the line tool. So the width is typically taken at an approximation of the quarter of the length. Once again, we use no scale bar and a length over width ratio. Next slide, please. Now for method 4, we decided to try a different approach. We took the length from the top of the villi to the bottom of the crypt, but instead of the width measurement, we took the length of the crypt. Our ratio this time was the length of the villi over the length of the crypt. Next slide, please. The last method we attempted essentially combines all the other methods with a few changes. We did use a scale bar of 50 micron for these measurements. We measured the length of the villi from the top of the villi to the bottom of the villi as opposed to the bottom of the crypt, and we took the width at the halfway point since there was no longer a risk of it being taken in the crypt. We also measured the length of the crypt. Now we were able to have 2 ratios from these measurements. We took a length over width ratio as well as a length of the villi over length of the crypt ratio. Next slide, please. Each method essentially built on the previous one. We use the errors or find areas of improvement to try testing in a new way. At the moment, the most we can do is identify a general range of ratios across the images for each method, which can be seen in the table. However, we're unable to draw any conclusions on the data at this time. Next slide, please. Our next steps include finalizing our method of measurement and collecting more data. We are also trying to develop a way to count the neutrophils or cells infiltrating the lamina propria using an image such as the one on the slide. And in the lab, they are also trying to work on a way of improving the histology images. On the next slide will be my references and then my acknowledgements. So I'd like to thank Dr. Lowe for mentoring me and giving me the opportunity to help in the study, as well as the graduate students in his lab for all their help. I would also like to thank the Rise and Persist faculty for making this possible: Jennifer Coplier, Jaren Paschke, Dr. Nugent, Dr. Wessler, my peer mentor Gina, the rest of the Persist cohort, Nii Tran, Marissa Carranza, Alex Cortez, and as well as the NSF for funding us. Thank you, and thank you so much for listening.
Right, thank you very much, Natalie. Very well done, great presentation.
Thank you.
Okay, it looks like you, happen to already have a question here from Marissa. She stated the unbiased measurement sounds like a great idea. Was this only possible because of the shift to virtual?
I believe so, because we were trying to figure out what I would be working on for the study. I think initially I would have been in the lab helping them take the images, but yeah, I think it was mostly inspired by the virtual shift.
All right, and it looks like you got a virtual seal of approval from your PI, Dr. Lowe. Thank you for tuning in. All right, we will go ahead and move on to our next presenter. Thank you very much, Natalie. And now we are starting the poster presentation. So the slight difference of poster presentation, students were requested to create an actual poster that would be similar to what you'd see if it was printed up in an actual symposium. Students have utilized a new zooming feature within PowerPoint where we will focus on certain specific sections of their poster. And timing wise is going to be 8 minutes total. So 5 to 6 minute presentation with about 2 minutes of questions. So our first speaker will be, first poster speaker will be Mr. Jonah Brunson. If you have an opportunity, please go ahead and turn on your camera. Thank you, Jonah. Your poster is on the screen here and I will leave it to you. Take it away.
Right. Are you able to hear me all right?
We can hear you fine.
Right. Well, sounds good. So hello, my name is Jonah Brunson. I'm a transfer student entering my 3rd year at UCR, and I'll be discussing the flash pyrolysis mechanism of trimethylchlorosilane, as well as a very brief explanation of its relation to silicon carbide thin film production via chemical vapor deposition, or CVD. But first, I think it would be appropriate to answer the question, why is this project important? So first, in a more general sense, the data produced through this project can be used to expand the knowledge of gas phase pyrolysis kinetics and create more complete models for related research. But more specifically, by understanding the decomposition channels of trimethylchlorosilane, its value as a silicon carbide precursor can be evaluated. Silicon carbide is a valuable compound that has been found to provide excellent resistance to thermal shock, as well as high maximum current density, leading to its use in semiconductor processing applications via CVD. So to analyze trimethylchlorosilane's decomposition, flash pyrolysis with vacuum ultraviolet photoionization time of flight mass spectrometry was used along with density functional theory calculations to support the 3 initiation channels. Thank you. So this image is actually taken from Kevin Weber's doctoral thesis, but a similar apparatus was used in this experiment. So starting from the left side of the image, a pulse valve is used to introduce trimethylchlorosilane along with helium as an inert carrier gas at supersonic speeds. The gases then enter the silicon carbide heating nozzle, which is heated by running an electrical current through the 2 heater electrodes. And then upon exiting the heating nozzle, the unreacted precursor, along with any products formed, are cooled by supersonic expansion and subsequently focused into a molecular beam by the skimmer, which is that sort of hollow cone shape with just a small opening at the tip. From here, the molecular beam enters the ionization chamber and is hit by a 118 nanometer laser, which provides 10.51 electron volts of energy. Any molecules that have an ionization energy that is at or beneath this provided energy are ionized and then gain a positive formal charge. This positive formal charge allows them to be accelerated by electric fields generated by the blue plates shown in the diagram. And as the particles accelerate towards the detector, they are separated by their mass, as heavier particles will move slower than the lighter particles. From the data generated by this detector, a mass spectra can be generated using additional software, and in the case of this project, we used Origin. If you can go and exit the image just for a second. Thank you. So some key characteristics of this method would be the supersonic molecular beam and the short residence time in the heating nozzle. Radical species are highly reactive due to the unpaired electron in their valence shell, and so only exist for a fraction of a second. By using this method, radical intermediates that would normally go undetected in other techniques such as gas chromatography can be observed, and a more complete model can be constructed. You can go ahead and just click on that top image in the results section. So here we have the stacked mass spectra of trimethylchlorosilane at temperatures ranging from 295 to 1370 degrees Kelvin, with the mass to charge ratio represented on the x-axis and the relative abundance displayed on the y-axis. So if you can see at the right, we see peaks representing the unreacted parent molecule trimethylchlorosilane at m/z equals 108 and 110. There are two peaks at approximately a 3-to-1 ratio because of the natural isotopic abundances of chlorine 35 and 37 respectively. This same ratio can be seen in the peaks at m/z equals 93 and 95, which correspond to the loss of a methyl radical, and in the peak at m/z equals 73 corresponds to the loss of a chlorine atom. The peaks at m/z equals 73, 93, and 95 are most likely the result of photoionization fragmentation, which occurs when the laser provides enough energy to break chemical bonds within the parent molecule. These peaks remain fairly constant until approximately 1270 degrees Kelvin, where peaks appeared at m/z equals 15, 28, and 42, which correspond to a methyl radical, a lone silicon atom, and SiCH2, indicating the beginning of thermal decomposition. At approximately 1310 degrees Kelvin, a peak appeared at m/z equals 68, which corresponds to the mass of SiC3H4, a secondary reaction product that can also be observed in previous research by Liu et al. This peak was observed to grow at the fastest rate, indicating that this particular reaction channel is the most favorable. And finally, also at approximately 1370 degrees Kelvin, a peak at m/z equals 40 appeared, which corresponds to silicon carbide. If you can go ahead and just exit the image for just a moment. And then you can go and just click on that image in the potential energy diagrams. Thank you. So this figure is a potential energy diagram that provides insight into the energy barriers for the possible initiation channels. So the calculations also provide further support to the mass spectra by indicating that the elimination of HCl is the most energetically favorable, followed by methyl radical loss and loss of a chlorine atom. The energy barrier for the loss of methyl chloride is also displayed. However, methyl chloride's ionization energy lies above the energy provided by the 118 nanometer laser, and so would not be able to be observed. So this channel is not investigated any further. If you can go and exit the image. So after analyzing the mass spectra, performing the density functional theory calculations, and reading some previous related research, I was able to create the proposed mechanism shown here. If you can go and click that image in the discussion portion. Trimethylchlorosilane appeared to have decomposed via three main channels: the elimination of HCl, loss of methyl radical, or loss of a chlorine atom. As displayed by these arrows, you can kind of see the reaction pathways from each product to the next. Sorry about that. Temperatures above 1310 degrees Kelvin, it was observed that the secondary reaction channel forming m/z equals 68 shown at the top right of this figure became increasingly prevalent, which was reflected in the generated mass spectra, and this pathway has also been studied in other projects. If you can go ahead and just exit the image. So that actually concludes my presentation. I would actually like to take these final moments to thank Dr. Jingsong Zhang and Quanliang Xiao, who guided me through this experience and exposed me to fascinating computational and theoretical concepts, CNAS alumni, RISE donors, and the National Science Foundation for the funding that allowed us to perform this research, and also my peer mentors and the rest of the RISE faculty and staff that worked to make this an enriching experience despite being in an online format. Thank you, and I'll open the floor for questions.
All right, thank you very much, Jonah. Great presentation. Okay, does anybody happen to have any questions for Jonah? Feel free to type in the webinar chat. I know it looks like we may have run just right on time, so we'll probably go ahead and start with our next one as well. Dr. Zhang has given his virtual seal approval to Jonah. Again, thank you very much. So looks like Zara, we have your presentation. No, that looks like, yeah, that's going to be the last poster. We kind of have one missing here. Just be 1 second. There we go. All right, so you can go ahead and take it away. Marissa will be supporting you in the back.
Thank you. Hi, I'm Zara Saeed, and this summer I worked in Dr. Jessica Purcell's lab looking at task partitioning in ants. So many social insect species allocate specific tasks to workers of different morphologies or ages, and this behavior is hypothesized to increase the efficiency of the colony. We used one species of Formica ant who specialized in tasks based on their body size. Large workers specialize in protein foraging and nest building, and small workers specialize in sugar water collection. And the aim of this study is to assess whether ant workers perform better at the tasks in which they specialize. We also observed the frequency and duration of trophallaxis, which is a food sharing behavior that allows ants to find food sources. So if we could move on to the methods section, please. Okay. So we selected groups of small and large Formica ants and placed them into separate artificial nest boxes along with a control group of small and large ants, so a mixed colony. And then the ability of the workers to perform the nest building and sugar water collection tasks was assessed. And then the part of this study that I contributed to was watching videos of ants and counting the number of trips that they took into the task compartments. And I also recorded and observed the frequency and duration of these trophallaxis food sharing interactions. And this is just a picture of the artificial nest box setup. So in the middle compartment, there's the nest building task. And in the big compartment, it's just a tube of water for the ants to rest in. So if we could move on to the results section. If we could zoom in on that. So we used a program called R to analyze the data that we collected. And when looking at the activity levels for nest building and sugar water collection, figure A shows the activity for nest building and task colonies, the nest building task colonies, and there was no significant difference between the large and small worker activity, but the mixed colonies had slightly lower activity levels. However, the work that the grad student that I work under did, was assessing the ability of the ants to perform the tasks, and large workers actually collected more nest material than the small workers. And then if we're moving on to the sugar water collection task, which is figure B, there was no statistically significant difference between the activity levels of the large, mixed, and small colonies. However, there is a slightly smaller activity level for the smaller ants, as you can see there, and in their natural environment, due to their size, they could be better at performing the sugar water collection because they are a small size, that they're similar size to aphids, and they collect the sugar water from them, so they would be able to access them easier than large workers. And so if we could move on to the next set of figures. So this is for the trophallaxis data, and looking at figure A, the number of trophallaxis interactions for the sugar water collection task was much higher than the nest building task colonies, and this can be explained by the fact that this food sharing behavior allows for the recruitment of other individuals so that they may find food sources a little bit easier and faster, so that it will increase efficiency overall. And for the duration of the trophallaxis interactions, which is shown in figure B, this figure is a violin plot, and the width of the plot indicates the probability of the data to show up in that area, and the line in the middle is the median. So for the duration, it was also longer in the sugar water collection task than the nest building task. So if we can move out to the overall view of the poster. So these preliminary results suggest that large workers more efficiently perform their allocated tasks, and they had similar activity levels to the other treatments, but collected more nest material, which is not shown because this was not part of my work on this project. Large and small workers collected similar amounts of sugar water, which is also not shown. Although there is no significant difference in activity levels while collecting sugar water, the data does show lower activity levels for small workers, which may be biologically relevant in nature, as I previously mentioned, with their size being an advantage when interacting with aphids and climbing up trees to access them. Overall, our results suggest that social insect colonies may take advantage of the different abilities of individual workers in order to increase the efficiency of the colony through task specialization. And further studies could build upon this and investigate further on a wider scale using several species of Formica and other social insects. So I would like to take the time to acknowledge that this project was supported by Dr. Jessica Purcell's 6 Legs program and funded by the USDA NIFA grant. And I would also like to thank me and Marissa and all the peer mentors that guided us this summer. And I'd also like to give a special thanks to Dr. Purcell, Mari West, Madison Sankovitz, and my fellow RISE students, Stephanie Ola and Shakira Palmer, for their help this summer. And I would now like to open up for any questions that you may have.
All right. Thank you very much, Zara. Great presentation, right on time too. So we do have a couple minutes for questions. If anybody would like to ask, please go ahead, type into the webinar chat, or you can raise your hand, your virtual hand.
So I actually analyzed, so the question in the chat says, how many hours of video do you think you helped analyze? We looked at the first hour of footage for several different colonies. So I'd probably say around like at least like 10 hours at the minimum, but probably a lot more than that, because I had to end up watching the videos like 3 times to look at the activity in each compartment and count how many times ants went in and out of those compartments. So yeah, and there's another question that says, what sort of future projects could help develop this and this work? Like I stated, so looking at multiple different species of Formica and looking at a lot of different other social insect species like bees and termites, they could also be used for a study like this.
Thank you very much. All right, that was a great project. Thank you for the questions, everybody. So we will move on to our last poster presentation before our second break. We have a co-presenters, Ms. Kirsten Bailey, as well as Mr. Luis Velez. Your poster is on the screen now, so you both can take it away.
All right, thank you, Nee. Okay, so I'm Kirsten Bailey. I'm an incoming microbiology transfer.
And my name is Luis. I am an incoming cellular, molecular, and developmental biologist.
And we've had the opportunity to work in Dr. Carrie Mock's lab this summer. Our research looked into the practical uses of plant priming. Plant priming assists in activating
Sorry about that. Kirsten is back.
Sorry about that. Okay, can you hear me now?
No, we hear you perfectly.
Yeah.
Okay, sorry about that. I don't know what that was. Okay, so we were studying insects who can possibly infect the plants or vectors. These vectors have the ability to lower crop yields of certain fruit producing plants. And in the US alone, crop losses can cost well over $1 billion every year, caused by viral agents transmitted by these vectors. So if plant priming was to be found effective, these methods can be an alternative to spraying pesticides, which can take a toll on surrounding areas and on the ecosystems of the areas where it's being sprayed. Are we still good, guys? Can you hear me? Yeah, okay. So to explore this, we used a synthetic phytohormonal analog to prime the melon plant defenses called acetbenzolar S-methyl, or ASM. We used aphids for our experimental vectors because they are known to feed on the melon plants, which we were using. And we monitored the plant wellness along with amounts of offspring that the aphids were going to produce. So to explain this experimental design a little bit more, there were 36 melon plants. These were cantaloupe plants, and they were grown on campus in the greenhouse. 4 of the plants were used to house the adult aphids, which were maintained in the lab. Adult aphids were used in order to have them reproduce when they were needed to. So the 2 remaining plants were going to be our infesting group. So on day 1, we sprayed our experimental treatment on what were known to be ASM positive plants, and we sprayed water on what were going to be our control plants. We gave 2 days for the plants to be in their prime stage, and during this time we monitored their health. So if we could zoom into the day 3, day 5 picture. And you can see we used a really general plant rating system because, you know, there are different things happening with 32 plants. And oh yeah, the plants were in 2 different places, my backyard and Luis's backyard. This is where we did the experiments. So in order to not harm the aphids when we were infesting the plants, we cut out pieces of leaf, which had 10 aphids on each piece, and with scalpels, and we used tweezers to carefully balance the leaf pieces on a broadleaf. And we can zoom out of the picture. After this point, we kept the plants well watered. The heat was extremely high the week that we did these experiments, and we counted the aphids on each control plant and each ASM positive plant on any given day. So we wanted to see the effects of honeydew or the sugar excretions given off by the aphids. So during this time, we set honeydew traps under densely populated leaves on each plant, and then we took these honeydew collections and turned it into a solution which we use for the behavioral assay in order to determine Argentine ant preference. And I will let Luis take it away with our results.
Okay, so if we can jump into the next slide. So this is the aphid population size. As Kirsten mentioned before, about 10 aphids were infested on each plant. And when looking at the graph, there isn't too much of a difference. On day 3, we had about a p-value of 0.75. And as for day 5, we had a p-value of 0.90. Okay, can we move over to the next slide? All right, and this graph is just showing the plant health rating system, as mentioned before from Kirsten. So as we can see here, there isn't too much of a difference. This was kind of used to determine the leaf quality. And as you can see, there's not much of a difference. The plants remained green the entire time. There wasn't too much modeling on the days that they were observed. Okay, and the next slide, please. And this is the graph that we use for the ant well plate test. So when comparing the water negative control to the ASM, there isn't too much of a difference, but when looking at the water negative control to the control honeydew, we see that a great portion visited the control honeydew, and then an even greater portion visited the sucrose positive control. Okay, and if I can have the next slide, please. To the discussion.
Oh, zoom. There's no zoom on it.
Okay. So statistically, there wasn't too much of a difference between both the ASM and the control plants. So unfortunately, the results failed to support the hypothesis. In terms of the next steps, where can we go from here? We could look at things such as the different environment, different weather conditions, the amount of aphids we infest the plants with, to see if we get a different outcome. And as for the plant wellness test, or the plant well test, we could use a higher concentration of honeydew to see if we get a more favoritism towards a certain plate. And I will leave the references and acknowledgements to Kirsten.
Okay, so we would really like to thank our PI, Dr. Mock, for her guidance, Dr. Purcell in the Six Legs grant, And we also would like to thank the USDA and the California Melon Research Board, Jamie Kenny for her previous research done, and Nee, Marissa, David and Nate, our peer mentors, as well as Isaiah, Gina, Karina, and also Syngenta for providing the ASM compound that we were studying. Thank you.
All right. Thank you very much, you two. Great presentation. We've run pretty much right at the time. If anyone happens to have any questions, feel free to type into the chat, and perhaps Kirsten and Luis can answer them virtually. But in any case, we will begin our 5-minute break right now. This is our last break before the very last session of the RISE Symposium. So again, thank you very much, Kirsten and Luis. Great job. Welcome back, everybody. We have our next presenter up, and just be a moment here while Marissa gets situated with the next slide. But thank you for coming back. Breaks always seem short to me, but we are on the home stretch now. So our next presenter is Ms. Megan Woods. Your poster is on the screen, and we look forward to hearing your presentation. So take it away.
I'm muted. Hi, everyone. My name is Megan Woods. I'm part of Dr. Lynn's lab. My research project this summer was time-dependent density functional theory investigation of UV-Vis spectra of isomeric compounds in brown carbon aerosols. So if you could please click on the background slide for me. To give some context of what that actually means, brown carbon aerosols are fine organic particles emitted from large burning biomass events, such as the California wildfires that we're currently experiencing. These aerosols have recently gained attention for their light-absorbing properties, which may influence climate forcing. Brown carbon aerosols are also currently understudied and lack readily available standards. To overcome this barrier, we actually utilize time-dependent density functional theory, or TDDFT, which is a computational-based method that facilitates overcoming this challenge. TDDFT projects theoretical UV-Vis spectra, so we may observe the brown carbon aerosol properties. In addition to the lack of standards readily available, there's a question of isomerization in the aerosols. The varying spatial arrangements of the molecules impact the optical properties, which creates a challenge of classifying the brown carbon aerosols. If you could move to the next section, please. Objective. Thank you. So what I worked on particularly this summer was to overcome this challenge. To do so, I calculated which isomer was most likely to emerge from the possible variations before running it through TDDFT. Overall, I was able to use these methods to reliably project a theoretical spectra. If you refer to Table 1, these are the compounds that I chose to display today. These molecules have functional groups that are often common brown carbon aerosols, and they also have a variety of isomers available. If you could go back, please. Thank you. And if you could hit methods. Thank you so much. So in order to analyze these molecules, what I first did was constructed the isomers using Gauss View 6. Then I optimized the structures using Gaussian 16. To determine which isomer would be the major product, I performed frequency analysis using Gaussian 16. From frequency analysis, I was able to observe the Gibbs free energy determine which molecule would be the major product. After determining which molecule would be the major product, I then ran the molecules through TDDFT and extracted the data through Gauss View 6 to plot a theoretical spectra. And if you see in section B of analytical methods, the theoretical spectrum is actually the summation of each peak profile. Could you please click back and then hit results, please? The theoretical spectrum ended up looking something like this. If you see in figure 1A in comparison to figure 1B, the trans configuration in comparison to the cis configuration, there's actually an increased absorption with the 300 to 700 nanometer spectrum in the trans isomer compared to the cis isomer. This is due to the increased stability of the molecule in comparison to the cis configuration. There's actually an increase in absorption between the 300 and 700 nanometer range, which is pertinent to our research because that's the range of which solar radiation occurs in the troposphere. Similarly, in figure C and figure D, there is an increased amount of in-plane intramolecular hydrogen bonding. This increases the stability of the molecule, which ends up resulting as the major product. If you could please hit back. Thank you so much. So in conclusion, we were able to observe a change in optical properties with the variation of isomers. Isomers with configurations with transorientation to intramolecular bonding increase the molecule stability. And as a result, we were able to determine the major products in comparison of the isomers that lack these characteristics. Molecules with increased stability had an increased absorption within the 300 and 700 nanometer range. And from this established background, we're able to pursue our work by working on building a connection between the chemical composition of the optical properties of the aerosols, as well as work on improving the full width half maximum estimation of the TDDFT theoretical spectra. We will also be able to pursue development of an increased understanding between the absorption spectra and climate forcing in order to implement these parameters in climate models. I would like to acknowledge Dr. James Sixman Fertile Futures Grant for funding offered by USDA, the USR Summer Bridge to Research Program, and special thanks to my graduate mentor, Kunpeng Chang, and Professor Lin, as well as all of the coordinators of the RISE program. And now I will open up to questions.
Thank you very much, Megan. Great presentation. If anybody has any questions, please feel free to type in the webinar chat here or to raise your virtual hand. Okay. Like maybe some people may be taking time to type if they would like.
Yeah.
And yeah, obviously having this type of research is extremely relevant even now, so given the current situation here. What was one of the more interesting aspects of doing this type of work and learning about it over the summer?
Well, I had no background in computational chemistry beforehand, so there was definitely a learning curve to overcome, but I was able to learn a lot of really interesting analytical methods. Also, I learned a little coding, which I had no background in, and so those graphs and results I all coded. And so that was a really interesting skill set to learn.
All right. Looks like you have the virtual seal of approval from Dr. Lin. She has stated, fantastic job. And a quick question from Mr. Kendrick Davis. Are there any other types of aerosols you are interested in studying?
Thank you, Dr. Lin. And currently I'm interested in pursuing just brown carbon aerosols, but I'd also be interested in looking into black carbon aerosols as well further along the road.
All right, thank you very much.
Awesome, thank you so much.
All right. We'll be moving on to our next presenter, Mr. Brian Zhang. Thank you very much for turning on your camera. Your poster is on the board, so please take it away.
Hello, everyone. My name is Brian, and I'm a rising third-year cellular, molecular, and developmental biology major, and currently continue research in Dr. Dahesh's lab. My project this summer is on the Golgi localized protein rewires stress response transcriptional network. In extreme and unpredictable environmental conditions, plants evolve the general stress response network by rewiring transcriptional networks to maintain cellular homeostasis and organismal integrity in response to abiotic or biotic stress. The lab has previously identified a cis element located in 30% of promoters of stress-specific genes designated as rapid stress response element, or RSRE for short. Genetic screen was employed using transgenic plant expressing RSRE luciferase, which is a bioluminescence enzyme. If we take a closer look in the background, for figure 1, comparing wild type on the left and the mutant with consecutively activated RSRE, named CAR16 on the right, you can see the luciferase expression on CAR16 mutant has increased compared to wild type indicated activated RSRE. In figure 2, the CAR16 protein was identified to be located to a Golgi localized protein. And in the bottom right corner of figure 3, you can see that CAR16 mutant in 3-day darkness treatment displayed an early senescence phenotype with much more yellow leaves compared to wild type in 3 days darkness treatment. Can we zoom out? My research question for this summer was, what are the differently expressed genes in CAR16 mutant compared to wild type in 3 days darkness treatment? To answer this question, RNA sequence analysis was performed on wild type and CAR16 mutant subjected to 0 and 3 days darkness treatment. More about my methods for RNA sequence analysis. Quality control was performed on the raw sequence reads using FastQC. The alignments were counted using, the alignments of the raw sequence reads were done mapped to the reference sequence of Arabidopsis thalia, tier 10 using bowtie 2. Then the alignments were counted using featured counts to produce a raw counts table. The raw counts were then used for differential express gene analysis done in R using the package dseq2. The results comparing CAR16 and the wild type were then filtered for p-adjusted value less than 0.05 and log fold chain greater than 1 and less than -1. Then the filter results were used for GoTerm and KEGG pathway analysis, which were done in R using the cluster profiler package. Now we take a closer look at figure 4 and 5. Figure 4 is the Venn diagram comparing the up and down regulated genes between wild type and CAR16 3 days darkness treatment. We identified 1,558 unique upregulated genes and 1,124 unique downregulated genes in CAR16 mutants in 3 days darkness treatment. In figure 5 is the heat map of all the log fold chains for all the samples, with the red shades being associated with a log 2-fold chain between 0 and 4, and the blue shade being a log 2-fold chain from 0 to -4. As you can see, there's a cluster of genes on the top right corner in the upregulated in CAR16 mutant in 3 days darkness treatment compared to wild type in 3 days darkness treatment. Now we look at figures 6 and 7 for the enriched GoTerm and enriched KEGG pathway analysis. Here is the top significant GoTerms and pathways, which were then subdivided into activated or suppressed. For the enriched pathway in figure 7, the most significant activated pathway was found to be the protein processing in the endoplasmic reticulum, or ER, for CAR16 compared to wild type in 3 days darkness treatment. We focused on the significantly activated pathway due to immune being a constitutively activated RSRE mutant. To visualize the results of the pathway analysis, let's take a closer look at figure 8 and 9. We focused on the protein processing in the ER and the associated snares interaction in vesicular transport. The proteins highlighted indicated the genes that were significantly upregulated when we cross-referenced it with the results table produced in the differentially expressed gene analysis. Figure 8, the snares interaction for vesicular transport, BET1 was found to have a 2-fold increase over wild type, and for figure 9, the BIP family of proteins in protein processing in ER was found to have a 3 to 4-fold increase over wild type. If we can zoom out. For conclusions and discussions, we identified 1,558 upregulated genes and 1,124 downregulated genes in our CAR16 3 days darkness treatment. Protein processing in the ER was identified as a significantly activated pathway in our CAR16 mutant. The lumen binding protein BIP family was amongst the most significantly upregulated groups known to be involved in protein processing in the ER pathway, approximately 3 to 4-fold increase. BIP1 has been shown to bind to stress sensor transcriptional factor BZIP28, and in presence of stress dissociates to activate an unfolded protein response. And for the next steps of the project involves clustering the heat map of the log 2-fold chain by GoTerms. Future experiments investigating genes identified in KEGG pathway analysis, especially ones in protein processing in ER, determine the nature and mode of action of genes involved in general stress response, determine the molecular mechanism by which CAR16 mediates transcriptional rewiring in response to stress. And here's my following references for my project. And for acknowledgements, I would like to give a big thank you to both my mentor, Jean, and Dr. Dahash for their powerful support and mentorship. I would also like to thank everyone in Dahash Lab. Thank you to Dr. Sue Wessler, Dr. Connie Nugent, and everyone in CNAS Persist and CNAS Scholars for making this research possible. Thank you for the National Science Foundation for funding this project. And I'm opening this up for any questions.
Thank you very much, Brian. Great job. If anybody has any questions, please type into the chat or raise your virtual hand. We do have time for a question. And again, really good presentation. Really like your transitions there. All right, looks like I'm not sure. Some people are still thinking about a question or taking time to type it up, but you do look good today. Thanks for dressing up.
No problem.
All right. We're at the 46 mark here. So looks like we will be moving on to our next set of presenters. This is actually going to be a committee group with 4 wonderful students. I will allow them or let them introduce themselves. See Mr. Andreas taking a sip of water as if he didn't have enough through your intro video. He was our sole student who actually took on the challenge and dove underwater and signed part of his intro. So great job, by the way. Glad we're able to see it because I think it skipped out on day 1. But anyway, your posters are up. You all look beautiful. So I will let you guys take it away.
Thank you, Nee, and good afternoon, everybody. We are back with more information on our favorite plants, rice, Arabidopsis, and tobacco. To reiterate, me and my teammates, Carolina, Andreas, and Eric, had the opportunity to work under Dr. Nagel's lab, where we researched genes that are regulated by the circadian clock and how they respond to heat stress within rice. Rice, being a globally consumed crop, was chosen as the focal point due to climate change having detrimental effects on its productivity. The circadian clock is a mechanism that plants can use to deal with a changing environment. Our experiment is a continuation of previous data sourced by Dr. Nagel's team, where they explored gene expressions for Arabidopsis. And our goal was to use our Arabidopsis heat response genes to see if we could be able to identify them within rice, and from there to determine if the circadian expression was conserved across the species. As to how we accomplished that, we will zoom into the methods portion, which has a flow chart summarizing what we did. With 3 plants in total, our starting point was with Arabidopsis. Our team identified the rice gene of interest, amplified it, cloned it into bacteria via colony PCR, checked the sequence to make sure that it was actually there, and then cloned it into a vector, which was then capable of infiltrating the tobacco plant. From there, we were able to analyze its rhythmic response to light and generate graphs of that data. As for the products we produced, Carolina will discuss that more in the top portion of the results number 1.
Thank you, Kiani. So can we zoom out, please? And zoom into the top portion, results. Okay, so as Kiani previously mentioned, we are going to begin by identifying and cloning the rice gene promoter region into a luciferase vector called P-Flash. The horizontal image at the top represents an image analysis of a gene structure diagram. Here you can see highlighted a simplified visualization of what you may see when looking further into one of our rice genome sequences for our project. Here you can also see highlighted in blue the location of the forward and reverse primers that we used in our project. Here also highlighted in green is the 1 kb promoter region upstream from our gene of interest. Moving to the image on the right, this is an image taken of our P-Flash vector map, which is used throughout this project. After gathering our rice sequences, we are then able to annotate it in a publicly available database called BenchLink. Once we have our vector map laid out, we are able to identify where our forward and reverse primers are, our 1 kb promoter region, and the luciferase enzyme are located in our P-Flash vector. This is the same vector we use to align the sequences after we sequence them in the plasmid and then to verify that the promoter region was indeed correct. We are then able to run a PCR on an agarose gel, which are the 2 darker images towards the bottom. The expected size is 1 kb, and we are able to confirm that our products did indeed have the predicted length due to the 1 kb DNA ladder ran on the same agarose gel for reference. And now I'll pass it on to Eric to further discuss our results.
Thank you, Carolina. So if I could be directed to the bottom half of results 1, please. So as Carolina was mentioning, there is a certain kind of bacteria, specifically agrobacteria, that can combine with plants, and as seen to the top left image corner, those are our tobacco leaves, and the image beside it shows some wet spots on the leaves indicating that we had infiltrated the luciferase. And the image next to that one shows the illuminating spots on the leaves of the tobacco plant, indicating a successful integration of luciferase. If you wanted to look at the graphs below those pictures, the images show the oscillations of the rice genes, showing promising results of the circadian clock working as the internal mechanism as genes were under constant light. And the graph below those, the difference between those graphs and the ones below are that the rice and Arabidopsis genes were exposed to light and dark over 3 days in a 12-hour period. The graphs were made from a publicly available diurnal database, and as you can see in these graphs, there are some sort of oscillations, but if you wanted to see a prime example of the key difference between rice and Arabidopsis is rice on the bottom left, you see one peak in each oscillation, whereas in Arabidopsis, you see 2 peaks for each oscillation on the blue line. So to further explain, I will take it on or leave it to Andres to keep explaining the second part of the results.
All right, thank you, Eric. If you can zoom out, please. And zoom into results and conclusions too. Now, this is the bioinformatic approach to our research where we want to determine whether the expression of our genes changes in response to other abiotic stresses. Using the publicly available resource, ePlant, we obtained the expression values of our genes in cold and drought conditions. We found that our genes in Arabidopsis, the 2 top graphs, have a more significant expression under cold conditions, as denoted by the asterisks. While in rice, the bottom graph, our genes have significant expression under both drought and cold conditions, making these genes good targets for further investigation. However, the time rate of expression and the response of abiotic stresses differs for most of our genes between the 2 species. We can zoom out now. The next steps for this research is to first finish cloning our 2 other genes, test the response of our rice genes to heat stress in the morning and afternoon time frame, and eventually create rice lines that overexpress their genes to see whether the plants are more tolerant to abiotic stress. At this time, I'd like to acknowledge and thank our PI, Dr. Don Nagel, the Nagel team, RISE coordinators, and our peer mentors, as well as the National Science Foundation for providing the funding to our research project. Thank you everyone for listening, and if anyone has any questions, we'll be answering them at this time.
Great job, everybody. Thank you very much for presenting posters, your second presentation of this entire group. Looks like you do have a question. Do you have any hypothesis on the differences observed for gene oscillations, the timing of peak of expression between Arabidopsis and rice?
I'll go ahead and take this question. So our main hypothesis was that, it wasn't really much of the hypothesis, it's more like what we were trying to observe is we wanted to see if the circadian expression of these heat-tolerant genes found in Arabidopsis would be present in the rice genes that we had cloned and put into the tobacco plant. So first, that's the one thing that we wanted to make sure was actually present. So we put it under constant light to see how the different oscillations would be and if they matched up to the same ones of that of Arabidopsis. And that's what Eric was mentioning down below in the middle graphs underneath where the tobacco infiltration is. That's what they show. And the reason why we needed to do it in constant light first is that it helped us see what the circadian expressions would be in just that. It was more of a constant control for us. So that way we can make sure that the expression was actually being shown.
To kind of go off of what Keo was saying, in the bottom 2 graphs that Eric was talking about Arabidopsis and rice, those graphs actually had, in the diurnal database, they had a control. So in order for us to kind of have a control group of our data that we gathered from the ImageJ software, we had to create our own control, and that control was constant light.
All right, thank you very much. We have run out of time here. We're going to move on to the next presenter. You do have one question from Dr. Katie Burnett. You guys are welcome to respond to her directly through the chat. All right, so on to our next presenter, Ms. Adalia Orozco. If you can, please go ahead and turn on your camera. Thank you very much. Your poster is on the board here, so please take it away.
Okay, thank you for that introduction. Hi, hope you guys are all well. This summer I was able to work with Dr. Gachomo in the microbiology lab, remotely of course. And today I will be discussing the chemicals of emerging concern and the effects it has on the microbiome. So go ahead and moving on to the abstract in the left-hand corner. We will begin by discussing human population growth and how it has increased the demand for resources like water. Droughts are very, very common here in Southern California, and thus we have a very big lack of water because of it. And this has increased the need for using recycled water in agriculture and lawns, things like that. The benefits of using recycled water are it helps conserve water, increases water's capabilities, preserves potable water supplies. Overall, the recycled water methods are really good at filtering out biological hazards that can make us sick. And it's been a very good conservation effort. The problem with using recycled water is its inefficiency of filtering out chemicals, specifically chemicals of emerging concerns. The filtration process is, it's not small enough to get rid of these chemicals. And these are things that are very, very common, like pharmaceutical drugs such as acetaminophen, which is commonly known as Tylenol, and other things such as antibiotics, things that are, again, commonly used on a day-to-day basis amongst many people. Now, if we can go ahead and move on to the methods on the bottom left-hand corner, if you can zoom in on that. We are, okay, the study that we did was in order to determine the effects of acetaminophen on the microbial communities that we took soil samples of. If you go ahead and look at figure 1 in the center of the poster, you can see that soil DNA extraction was from potted plants irrigated with water containing CECs after 3 and 7 weeks. The total environmental DNA was then extracted, and we did a 2-step PCR method in order to build a DNA library. The first step in the PCR was used to amplify the 16S RNA bacterial gene. We then followed up with a DNA cleanup. The second step in the PCR used was to complete the illuminate adapters and increase concentration of DNA and purify it. We then followed up again with another DNA cleanup. DNA was analyzed to check for quality and quantity purposes. Then if everything looked good, we continued on and had it be sequenced in the illuminate machine, which is the MySeq. Now, at this point in time, this is when I joined the project and became an active member. I learned how to do DNA sequencing while using KIME software on my computer. Now, if we go ahead and move on to the results, we can see that in our results, we found that the DNA library prep was made successfully. If you look at figure 2, we can point out that the red arrow on the left was bad, it was smearing, and the white arrow on the right-hand side was a lot better, and we could continue on for sequencing. We measured the diversity in each sample using the Shannon-Wiener index, and error bars represented standard deviation in figure 3. We found that there was no significant difference in that each sample had the same amount of alpha diversity. Alpha diversity was the lowest in the acetaminophen treated samples at time point 2, which was 7 weeks. In figure 3, that would be the blue bar on the right-hand side. However, if we go ahead and look at figure 4 at the bottom, we saw major shifts in abundance with certain bacterial groups. Time point 1 and time point 2, treated with acetaminophen, showed gamma proteobacteria had a higher abundance than at the initial time point, which in figure 4 is the dark blue band towards the bottom. Now, time point 1 and time point 2, treated with acetaminophen, had a lower actinobacteria than the initial time point, which again in figure 4 would be like the gray band towards the center. And actinobacteria treated samples at time point 2 had a lower gematomonides abundance than any other samples, which again in figure 4 is going to be the light blue band. Now, in conclusion, acetaminophen may have some small effects on community diversity of the bacteria. Overall, some bacterial groups did experience a change. For example, gamma proteobacteria had an increase. This is a very large and diverse group, which can have a big effect on the soil and plant interactions. Gematimonides also had a shift, but since little is known about this group, just that they are present in most soil communities, we still need to do some more studies in order to understand the plant-soil interactions and implications. And on the other hand, actinobacteria class are a class of bacteria that are known to be antagonistic towards certain or many plant pathogens. We observed a decrease in this class, which could potentially affect the plant's ability to fight off pathogens. Our results point out that in the occurrence of CECs, the microbial communities of the soil do change in abundance and therefore can alter the microplant dynamic. More studies are necessary to determine more specific implications. I would like to take this time to make some acknowledgements to Dr. Gachomo, Nathan McLean, Melissa Gomez, Nitram, and the rest of the RISE program, and special thanks for funding from the Dr. James Sickman's Fertile Futures Grant, which is funded by the USDA. Without any of these people, I wouldn't have been able to learn and grow as a person, and I will take all of these new skills with me onto my future endeavors. Thank you very much. Are there any questions?
Thank you very much. Wonderful presentation. Does anybody happen to have any questions? Please type in the chat here, or you can raise your virtual hand. We'd love to hear anybody else's voice other than my own at this point. And again, really good job. Sorry your Zooms apparently did not work on here, but we all kind of got the gist of it. Abstracts are even available through the link that we provided earlier, which I'm going to share again. Okay. And what did you like most about your research for this particular summer? I know I've been hearing a lot about Dr. Gachoma's lab and work for you.
I think my favorite part was working with Nathan McLean. He taught me a lot of things about working on like a computer, and that was like the first thing I ever, or the first time I ever really did anything like that. I'm not really like techie, like, you know, when it comes to that kind of stuff, but I learned how to analyze data on the KIME software, and that was really, really interesting. It was a little challenging, but it was definitely like pretty cool to learn.
All right. Well, thank you very much. Great presentation again. We will be moving on to our next presenters.
Thank you.
All right, so we actually have two students presenting the next poster. We have Ms. Jessica Black as well as Mr. Lawrence. Thank you for turning on your cameras there. Your poster is on the board. And again, you guys can take it away. Thank you.
Thanks, Nee. So hi, everyone. My name is Lawrence. I am a third-year biology student.
And I'm Jessica Black. I'm a third-year neuroscience major.
And we are in Dr. Garland's lab, and our project is how mouse brain regions are affected by chronic voluntary exercise. So if you would zoom in for me, please. Thank you. For our project, we are working with graduate student Margaret Schmill on one of her dissertation projects. The project is actually a continuation of a previous dissertation experiment run by Zoe Thompson. The experiment focused on mice that had been bred for traits related to high voluntary wheel running, and these mice were part of a long-term project that focused on selecting lines of HR mice down about 66 generations at the start of this specific project. Now in the project, it was noted that the female mice tend to have a greater difference in their wheel revolutions compared to males when being studied on days 5 and 6. And this was the basis of the HR line of mice. So Zoe extracted the brains of these HR mice and found that there was an increase in the brain mass compared to the mice from the control group. Because of those results and other behavioral observations of the HR mice, they not only have an increased ability to run, but also potentially increased motivation. This project proposes that chronic voluntary wheel running may lead to a significant change within the brain that affects either the mesocortical limbic system and the locomotive system, or the locomotive systems. The specific regions that we are actually studying in this project are the basolateral amygdala and the hippocampus, and these regions were chosen on the basis that they are centers of emotion and memory, which might be a determining factor on the drive to exercise. So you can now zoom out, and I'll pass it over to Jessica, who will continue with our methods.
So for our methods, can you zoom in again, please? For our methods, we basically did them in 4 different stages. And the first stage was lab staff giving the mice 10 weeks of wheel access or 10 weeks without wheel access. And then the second stage was also done by lab staff, where they extracted the brains and imaged them. And before the imaging, they meso stained them, sliced them, and then the hippocampus was imaged at 20 times magnification, while the amygdala was done at 24 times magnification. And then the third stage is where Lawrence and I come in, and we used image J tracing to be able to trace our specific regions. And in order to do that, we also use the Allen Institute Mouse Brain Atlas, which is what you see in figure 3. And we compared the atlas to the images that we had in order to find our specific regions. And then we use an ITCN plugin to count the amount of cells within the area so that we could have the cell density. And then the fourth stage was data collection. And with this, we aligned the first set that we had traced with the second set. And then we just went through to see if there was a lack of uniformity, which Lawrence is going to talk about in our preliminary results.
All right, thank you. So if you could zoom out again, Marissa, and then zoom into the results. All right, so it's worth noting that these results are actually not conclusive and are in fact just the culmination of our efforts from these past few weeks. Our main purpose was just to limit human error as much as possible and to prepare our process for a greater sample size. Now, the data that we noticed that could be relevant regard that of the basolateral amygdala having a cell density range from about 0.0030 to 0.0035 cells per squared micrometer, and that the hippocampus has a range from about 0.0023 to 0.0026 cells per squared micrometer. This information can only represent about 10 brains from the whole 100 within the sample size, and we cannot yet determine if there's actually been any significant increase within these regions. But anyways, the process we underwent when minimizing human error involved us receiving about 10 brains labeled by their mouse IDs such as 69100, and then 10 brains labeled through 1 through 10, like brain 1, brain 2, brain 3. And these were actually just the same scans, just out of order. We were then told to trace our respective regions and compare our results in Excel between the mouse ID scans and the numbered scans. We would then create a graph shown like on the poster that juxtaposes both trace regions cell counts, and we could then see whether or not we have outliers that represent inaccurate tracings. And we could see here, represented by the purple dots on the graph, that these are actually outliers of our tracing. So using this information, we may then go back and figure out how to accurately trace amygdala and the hippocampus for the rest of our samples. And now we could zoom back out, and I'll pass it back over to Jessica.
So I'm going to be going over the conclusion and our discussion with you. And we thought, based on our preliminary results, we think that high-running mice are going to have a more evolved brain structure. And we came to this conclusion just based on the knowledge that with more stimuli, your brain becomes more complex. We also think that different regions won't have the same effects as other regions. And in the dissertation, the midbrain mass was a little bigger. And so that's where we came to that conclusion. And we also think that long-term exercise is going to be able to help combat the different effects that come with genetic related illness. An example that I can give you for this is maybe like the slowing or prevention of a degenerative brain disease like Alzheimer's or dementia, because if you exercise and let's say your hippocampus gets bigger, then when it deteriorates, it's going to take a little longer, so it'll slow down the process. And what's next for Lawrence and I is that we're going to continue the project, but with more brains and a larger sample size. We also plan on repeating the same process with other regions of the brain that might not have been covered yet in the dissertation. And we also want to look at how the research can be applied to people by looking at genetics, their physical activity level, and their level of motivation. And now Lawrence is going to go over our acknowledgements.
So thank you, Jess. I want to wrap it up by saying that these are our references shown here, and we want to thank the people involved with our program, especially Dr. Nugent, Dr. Wessler, Nee, Marissa, Gina, Jen and Jaren, and all of our friends within our Persist group, as well as everyone involved with the Scholars Program, Persist and RISE. And lastly, a thanks to our sponsors at the NSF who have made all our research possible. And if there are any, we would like to answer all your questions, and thank you all for listening.
All right, thank you very much, you two. Amazing presentation. You guys did a great job and great teamwork as well. All right, if anybody would like to ask any questions, please feel free to type in the webinar chat, or you can raise your virtual hand and ask it live. Again, while we give guests a minute to type up any potential questions. Yeah, really great job, and thanks for covering your next steps. It seems like you guys had a lot of great experience over the summer. It sounds like you probably had even more experience prior to the summer. Did both of you participate in Dr. Garden's lab during the regular academic year?
Yeah, so I got involved actually at the start of the year in January, and I've been in a couple times before the closures, but this is like my first time. The summer was the first time I'm working on this specific project, because I actually had another project like planned before everything closed, but because of that, it couldn't be possible.
And I basically had a similar experience to Lawrence, but I started, I think, around my spring quarter of my first year in the lab, and I had about 2 different projects that I was going to help with, but then everything closed, and so I wasn't able to. But this was a good opportunity to work on something.
All right. Well, we are glad to have both of you, and you had the opportunity to still continue the work and present today. All right. Thank you very much. So we will move on.
Thank you, Lee.
All right. Our very last presentation of not just today, but of the entire RISE Symposium. Last but not least, Mr. Chris Scatula. Thank you very much for turning on your camera. Your poster is on the board and Marissa will be supporting you, so take it away.
Okay, thank you, Nee. Good afternoon, everyone. I'm incredibly excited to be here with you all today. My name is Chris Gadula, and I'm an incoming second year majoring in biology. This summer, I was a part of Dr. Nicole Zernina's lab, where one of the main focuses is on embryonic stem cells. To give a little bit of background, embryonic stem cells are commonly known as pluripotent cells, meaning they can differentiate into the many cells of the body. We focused on the 2 pluripotent states that they can exist in, those states being naive and primed. The naive state is what the Zernina lab wanted to exploit due to the fact that it opens up a wide range of possibilities that the stem cell may differentiate into, unlike the shortlisted options in the primed state. In the prime state, the stem cell has decided on the pathway it will follow and has started to prepare itself to differentiate into different cells of the body. In order for embryonic stem cells to have effective biomedical applications, we must find a way to keep cells in a state free of epigenetic lineages and random differentiations, or in other terms, we want to find a way to keep the stem cells in a naive state. Throughout the past 8 weeks, the Zernina lab was studying a neurotrophic drug called 1D6, which was found to increase stem cell survival rate in feeder-dependent cultures by activating a receptor in the cell labeled as a TRK receptor, along with the fact that supposedly 1D6 is able to promote a naive state. The Zernina lab found that 1D6 activated the TRK receptor, but not another receptor called NGFR, which is activated by other neurotrophic drugs similar to 1D6. The receptor NGFR is supposed to promote cell apoptosis or cell death, which we do not want. Along with 1D6, we also use ROCK-I inhibitor because it is also known to help stem cell survival rate when the cells are passaged, which is a technique that enables an individual to keep cells alive and growing under cultured conditions for extended periods of time. Lastly, the third condition we had was a commercially available media called RESET or RSET media. We use this media because it is advertised to keep cells in the naive state. So for that purpose, theoretically, if the cells found in RESET were found to be naive, we can analyze the genes in those cells that are associated with the naive state and compare those lists of genes to the genes we would obtain from 1D6 and then conclude whether or not the cells cultured in 1D6 were naive or not. And if you could click on the graph on the bottom left for me, please. Through the use of RNA sequencing, we were able to determine the states that our embryonic stem cells were in. As stated, there was those 3 conditions, and there was also a control. Those samples were the 1D6, the RESET, the ROCK-I, and the control was grown in a different media known as M-teaser, and they also had no other drugs or inhibitors added to it. The cell samples, once harvested, were sequenced through a program called Illumina NextSeq. The files obtained from that program were then aligned with the human genome, which is labeled as HG38 for short, using a program called HiSat2. Through the use of this program, we were able to determine the genes in our samples that matched up with genes in the human genome. Next, the reads, or the amount of genes that aligned with the human genome, were put into a program called feature counts, which showed us how many times the genes in our samples aligned with the human genome. The genes that were differentially expressed, or in other words, up or down regulated, were identified with a program called EdgeR. Parameters were set in order to filter out lowly expressed genes that did not meet our criteria through use of statistical analysis, where we set a parameter known as a p-value. And if the p-value of our genes differentially expressed did not meet our criteria, they were excluded from our data. Lastly, through the use of a program called RStudio, we were able to create multiple plots and graphs that allowed us to determine the genes in our samples and their pluripotent state. And if you could click on my graphs in the middle, please. In this project, we found the most notable changes to be found in the samples cultured in rock eye. Thus, most of the graphs and visual figures are of those samples. As you can see in figure 1, it shows the gene ontology plot that showed the differentially expressed genes in rock eye. Based on the x-axis, we can determine the up and down regulation of the genes depending if the dots lay to the left or right of the 0. The y-axis shows a short explanation of their function. In our case, we saw that the genes for axon development and axonogenesis were found to be upregulated as they were to the right of 0, which are the 2 dots you see. This may indicate that these cells have chosen to start differentiating into neuronal cells. To the left of 0, we see a significant downregulation of genes for apoptosis and programmed cell death, which was what we expected because this means that rock eye was helping prolong the stem cell survival rate. In figure 2, it shows a smear plot of the samples in rock eye, which showed an upregulation of genes related to the prime state and a downregulation of genes associated with the naive state. In figure 3, it shows a heat map, which shows how closely related the samples were to our control group, the cells cultured in M-teaser media that I previously stated. The x-axis reveals the relation of the sample to the control group, which are labeled as clump, depending on how close you are to those samples. In figure 4, on the PCA plot on the bottom, please. With this, we can see that the reset and 1d6 samples were closely related to the control, and this was also evident in our data because we found that cells in 1d6 and reset did not show any genes differentially expressed for naive or prime. The most prominent change was found to be in the rock eye samples. In conclusion, analysis of our data showed that samples cultured in reset yielded no massive changes compared to the control and no change in pluripotency status. The 1d6 samples were also observed to closely resemble the control as indicated by the PCA plot and also showed neither no up or down regulation of genes associated with the naive state. The samples in rock eye downregulated apoptotic pathways as expected, and it also showed an upregulation of genes associated with the prime state and a downregulation of genes associated with the naive state. Therefore, although rock eye was able to reduce the apoptotic pathways, it showed cells being in the prime state, suggesting that rock eye is not suitable for embryonic stem cell maintenance and would deter them from contributing to different cell lineages. And if you can zoom out, please. Our future goals are to conduct more experiments of 1d6 by testing with different dosages and the length of the treatment time. Additionally, we would like to culture with other naive cell-defined media, which will be used as a comparison for the 1d6 treated cells. I'd like to thank Dr. Nicole Zerneden for giving me this unique opportunity to work with her lab. I would also like to thank her grad student Desiree Williams, as she greatly assisted me through the majority of this project, along with learning the concepts and materials being studied in the lab. I would also like to thank the RISE program and its donors, specifically Mr. Kenneth and Nancy Gray, along with the CNAS alumni that helped run this program and make it a great one, even through a virtual setting. Lastly, I'd like to thank all of you for attending the RISE Symposium and for attending my presentation, as well as the numerous others you may have listened to. I will now be open to any questions you may have.
Thank you very much, Chris. Great presentation and a great way to close out our symposia. So if anybody has any questions for Chris, please type in the chat. And that raise your hand option is always there. All right, I second that, Nate. Great way to end the event. Actually, we have more for you too, but we'll give people a moment. All right, and hopefully nobody should be leaving just yet, because we do have some general closing statements and announcements for everybody. All right. Well, thank you very much, Chris. It seems everybody may be overwhelmed, and also I know it is lunchtime. But appreciate all the hard work that you've done. Yeah, thanks for going to UCR and you know, presenting and dressing up today.
Thank you.
All right. Okay, so just to kind of close out here, we do have some final announcements that Ms. Marissa Kranza will share with you guys. Thanks for that. That was a little unexpected.
Hi, good afternoon, everybody. It's Marissa. I've been behind the scenes today, but This was my virtual applause. I'm sorry, I didn't mean to scare anybody, but I really think you guys deserved it. It was really great seeing your guys' professionalism and how much you really did learn, even though there was so much to overcome. You guys did so amazing. So I'm gonna play it one more time and hopefully you don't get scared this time, but your virtual applause is very much, I hope you receive it. So, please take a moment and really give yourself a pat on the shoulder. You know, you did an amazing job these last 3 days and we're very, very proud of you. And you should be proud of yourself. So as a reward for working so hard over these 8 weeks and for doing what many thought would be impossible, this virtual research experience, we have 2 things coming your way in the weeks to come. So you'll get a certificate of completion that we give to every single RISE researcher at the end of the symposium. We usually take a group picture and all that. So I want to encourage you that when you do receive this certificate, send us a picture. Put it on Instagram, share it with us. That way we can see your beautiful faces with your certificate of completion. Also, as a reward for finishing such a difficult task, we want to give you guys a memento of your time here this summer with the first ever RISE t-shirt. So this is the design, and that's why I sneakily asked all you guys your shirt sizes about 2 weeks ago. They should be coming in the mail soon, and this is your care package number 2. I hope you wear it with pride, and I hope you realize that what you've accomplished here was very special, and we're also very proud of you. So great job, everybody.
All right, thank you very much, Marissa. I do want to recognize somebody extremely special, and that is the person right in front of you right now. That is not me. So one thing I would like to mention again, in addition to all the faculty, the staff, students, the donors, everybody who has made RISE possible and what it is. Today happens to be Marissa's very last day. So Marissa has been going to school for nursing and has actually not only conferred her degree, but officially passed her licensing exam recently. So she will become a nurse at a hospital nearby, particularly possibly in the Inland Empire area. And today is actually her very last day working for UCR. So one thing I do want to mention is Marissa's been with us for over 6 years. She started off as a transfer student, sitting in the exact same shoes as the majority of everybody in RISE. She came in through the RISE program, went quickly through the ranks, graduated in 2 years, and she got hired immediately after and has been working ever since and making RISE what it is. I'll tell you right now, she's been the bedrock for our program, how it's become what it is today, and including this virtual setting. We could not have done it without her. So I wanted to send a very sincere thank you, and everybody else. Absolutely thank Marissa too. Again, she's been such an inspiration and support for all the students. She's extremely passionate of helping you guys, as you guys all know and see and have experienced yourselves. So again, thank you very much. And for the past 3 days, in case you guys did not know, Marissa has been coming into campus, having a little mini setup in her office. Yeah, so she's at UCR, that is Pierce Hall right there. And that's our old office where we would normally have been situated. And yeah, like I stated, great to have her on board. Exciting and bittersweet, but at the same time, more exciting to see her move along and go on with her future professional endeavors. So again, congratulations, Marissa. Thank you so much. And you can see everybody that you affected just even for this summer. Imagine there's been 80 students every single summer and then some.
Yeah, so I think it was so cool. Oh my God, I'm gonna start crying. But
You're good, you're good.
I just think it's so cool that my first day at UCR, it was as a RISE student. I didn't know anybody. And my last day is here with you guys, so it's just such a nice ending. I'm just so proud of you guys. I really hope I never meet any of you in my new job. Please don't come looking for me. Stay healthy, stay safe, wear your masks. And I really hope that you guys feel comfortable reaching out. I will always be there for anybody who needs any type of assistance. You guys are my passion. I love helping students, and I did not think I was gonna cry. But once again, RISE holds such a special place in my heart, and I'm so grateful, and you guys did an amazing job. So thanks, Nee, and everybody for your wonderful thoughts and comments in the chat. I really appreciate it, so thank you.
Thank you so much, Marissa. It's actually very appropriate. You are ending in the same room that the STEM Center used to be, and that's where you would sit and study and have your UCR career. And now it's your office, your former office. So in any case, thank you very, very much, everybody. It's been an amazing summer. Even we weren't sure how these 8 weeks were going to turn out, let alone even the symposia, but I'm glad we've come to an end. But again, this is only the beginning for most of you. We all hope to see you physically on campus at some point. Would love to have a gathering. Would love to honestly feed you guys, because that's what we are typically known for. We try to gather around a lot of food. But in any case, a lot of great comments, a lot of great supporters. Patricia, thank you, from Norco College, Dr. Nugent, all the faculty, lots of staff, all the peer mentor team. Really couldn't have done it without you guys as well. So again, thank you very much. This is our very last day of the symposia, and we will be signing off. So hopefully we'll see you guys in the near future. Bye, everybody.